mics at the beach, which is great. The meeting is being recorded, so if you don't want to have your face on the beach or wherever it might be, then if you want to turn off your video, that would be great. So my name is Chris Hardacre. I am the director of the UK Catalysis Hub at the moment. And we have various people from the UK Catalysis Hub as well as from the SuperGen Bioenergy Hub as well. Patricia, do you want to introduce yourself?
Yeah, sure. Sorry, I was just unmuting, Chris. I'm Patricia Thornley and I'm the director of the SuperGen Bioenergy Hub.
Great, and we thought, Patricia and I, so I'm in the Bioenergy Hub as well, and we thought it was a good idea to try and see where the linkages were between the UK Catalysis Hub and the bioenergy side of things, because there are lots of synergies between the two, but also there are some things that we don't obviously do in the Catalysis Hub, and there are things that the SuperGen Bioenergy Hub doesn't do either, and we wanted to work out where we might be able to interact a little bit more. Now, of course, originally this was supposed to be the first day of our summer conference for the Catalysis Hub, which we were all supposed to be having in Harwell, which I hope is sunnier than it is in Manchester, because it's not very sunny in Manchester at the moment, unsurprisingly. Well, not it's always sunny in Manchester, of course. Anyway, but we obviously can't, so we're doing it via Zoom. We have a number of presentations that are going to be done throughout the day, and I'll be chairing some of the sessions. Patricia, I think, is doing some of the others. So what we will do is that I'll give just a very brief presentation on the Catalysis Hub first, then I'll invite Patricia to do the same on the Bioenergy Hub, and then we will launch into the presentations. During the presentations, if you have any questions, if you can type them into the chat, and then whoever's chairing will then look at those and ask the question, and if there's any clarification needed, we'll ask you to unmute and so on. But if you could all mute yourselves for the moment, that would be very helpful. So I will just share my screen to start with, if I can do so. There we go. So I hope everyone can see that. I can make that. There we go. Can everyone see that okay? So the UK Catalysis Hub has been going for some time, and the idea of it was to establish a world-leading comprehensive coordinated programme of catalytic science and engineering in the UK. And we started this in phase 1 in 2013, and that ran for 5 years. And we've had a subsequent phase of funding from EPSRC that started in 2018. And it was really to enable the UK to regain and retain a world-leading position in catalysis. We had a world-leading position up until the sort of mid-80s, early 90s, and then gradually that dissipated with diversification and so on. And we've tried to refocus the community, and that has happened to a large extent. The first theme or first phase of the hub was associated with 5 scientific themes, and you can see those on the right-hand side: catalysis by design, energy, environmental catalysis, chemical transformations, and biotransformations and biocatalysis. And the whole idea was this was a collaborative activity combining theory and experimentation, also science and engineering across the UK. And you can see the map on the bottom right-hand corner with the 4 red dots associated with where the PIs for those grants are. So we had UCL, we had Bath, Cardiff and Manchester, and the yellow dot is where the research complex at Harwell is. But you can see also there were lots of other satellite universities that were involved in the projects as well. In total, we had something like 40 universities and over 100 academics being involved in the project. And in terms of our successes to date, we built up a very strong relationship with the central facilities, the Diamond Light Source, ISIS, as well as the central laser facility. We developed our early career researchers. We had a strong push to have gender balance. We developed the Harwell campus in terms of a hub, and we had a very vibrant scientific program over those sort of first phase, with 82 projects being funded. And I think now, Josie will correct me, but I think it's something like 240 publications over that period of time. Phase 2, which is where we are now, is in 4 areas where there is a core activity, which is to provide the advocacy and leadership, as well as to do some science associated with the central facilities, mainly on data management and development of new in situ cells and so on. And then we have 3 main themes or science themes: optimizing, predicting and designing new catalysts, Catalysis, the water energy nexus, and the circular economy, Catalysis for sustainable manufacturing. What we do in the hub is we have funding which is initially for 2 years, and that's just coming to an end in the next sort of 6 months or so, where the original exemplar projects which we proposed to EPSRC were then funded. And then we had an open call for projects which actually ends, or the closing date is a week on Friday, which is inclusive and open to all. Effectively, we don't say all the people who contributed to the original proposal, they're the only people who can apply. Anybody can apply from across the UK. And the whole idea is it's supposed to be collaborative and multi-institutional, plus multidisciplinary. And that could be between theory and experimentation. It could be between science and engineering, or all 3. It could be different types of catalysis, etc. It could be life cycle analysis coupled with catalysis to give an idea. What we are doing is trying to promote us having industrial input into those projects as well. So we are encouraging that substantially. So in terms of what we're doing in phase 1, in the core, we have these 2 main projects. One is high throughput and improved sample environments for catalysis at central facilities. This is to do with lasers, neutrons, and X-rays. And then we have something to do with data analysis. As we go through to more high throughput data collection in terms of understanding our catalysts under in situ conditions, we've got to be able to do more high throughput data analysis and processing. And so these 2 are linked in trying to develop our ability to be able to take a lot more data and be able to analyze it and understand it in a lot more detail. But the overall hub core also organizes conferences, workshops, it organizes our social media and also our community development. Obviously, some of these things have now gone virtual, and we've been doing quite a lot with the community through webinars and so on, stakeholder engagement through the virtual scenarios now as well. In terms of Science 1, which is optimizing, predicting and designing new catalysts, the idea is to understand those processes at a molecular level, both from the theory and the experimental perspective. And we have, these are the exemplar projects, and I won't go through all of these, but you can see they develop things to do with operando synchrotron catalysis studies, development of neutron scattering, as well as laser facilities, as well as things like where catalysis may be used in medicine, which is work package 4, how it can be used in cascade processes, and developing computational modeling and so on. Catalysis, the Science 2 in the Catalysis Hub in phase 2 is about how water and energy are inextricably linked, and how we can valorize water and valorize things like the contaminants in water. And again, you can see here, there are 7 work packages. They're fairly self-explanatory. We do have a life cycle assessment associated with this as well, where we obviously want to be able to produce catalytic processes and solutions that enhance our sustainability rather than cause a problem with our sustainability. And then there's a modeling package which also underpins our understanding of many of these other activities. They're not all associated with wastewater. Some of them are about transformations in water and with water, and some of them are developing energy and fuels from wastewater. The last science package is Science 3, which is Catalysis for the circular economy and sustainable manufacturing. And this is really encompassed by this bit in italics, which is keeping the molecules in play. So in other words, using things like CO2 and plastics and so on, how we might use those and recycle those and develop those into feedstocks again. And so you can again see the list of those projects. Again, there are 7 exemplar projects, which again talk about things like making or valorizing CO2 by making carbon-carbon bonds. So this isn't just converting CO2 into methanol. This is it converting it into higher hydrocarbons, understanding oxidation processes. This one here that Arthur Garforth is involved in, and I am as well, is about how we do things with our plastics and how we can convert those into new feedstocks and recycle them more easily, as well as building sustainable polymer architectures, which Charlotte Williams is involved with, for example. You can see, again, this is the map of where all of the universities are that are associated with phase 2. As we have done with phase 1, we have 23 projects ongoing at the moment. We will increase those number of projects as we go through the next proposal round, but they are all multidisciplinary, they're all multi-institution. So it's not one postdoc, one academic, it's one postdoc, many academics, to be able to give us the cooperative nature of the projects. So that's the introduction to the Catalysis Hub. I'm happy to answer any questions that anybody may have. Let me try and expand my screen. So if I stop sharing, I can do that. Does anybody have any questions on the Catalysis Hub at all? I see nothing in the chat, so if not, then I will pass over to Patricia. I don't know whether you can share your screen, Patricia. Let me just double check. Ah, you can share your screen. You're on mute at the moment.
Can you hear me now?
Yes, we can hear you.
Right, okay, brilliant. Right, too many screens open. Okay, so yeah, I'm going to essentially give you a very similar presentation, but from the bioenergy side. So I said earlier, I'm the director of the SuperGen Bioenergy Hub. We have a very similar history, been in operation since 2012, with 2 funded periods. I guess we've got slightly further to run in our current funded period. So we are funded through until 2022 at the moment. So this is what the hub looks like. We look at bioenergy from a whole systems perspective, and we break that up into our topic group 1, resources, which is led by Ian Donison at Aberystwyth. Topic group 2 is the one that Chris co-leads, and that is led by Chris along with Jason Hallett at Imperial College and Tony Bridgewater here at Aston. And that's all about pre-treatment and conversion. I will come back to that in a moment, but just moving on, topic group 3 on vectors. So this is about what are we trying to turn our biomass resource into? Biomass resource comes in all shapes and forms. Chris has already mentioned wastewater. We often get involved with things like the residues from agricultural production or wood from forestry systems, as well as wastewater, sewage, sludge, and municipal waste. But it's a question of what it's useful to turn those into. And this is where I think catalysis is actually quite key, because many of the conversion systems that we run biologically, thermochemically, or catalytically give us fairly crude products. And often we're trying to fit something with a very specific existing specification, be that a gas for use in the UK gas network or an aviation fuel. And we often find that we are using techniques of catalytic upgrading as we go through these processes. And I think a more holistic approach where we looked at the entire conversion system, what we're starting with and what we're trying to get to, and do that in the most energy efficient and environmentally effective way makes much more sense. In order to evaluate that, we have, you will see on the far right, case studies. We have spent probably about a year of SuperGen consulting with the community on what systems could be materially significant for the UK in future. What are we likely to see massive implementation of when we look at our 2050 objectives, our biomass resources, and what we believe our future energy demand will be. And that's involved things like looking at how liquid fuels may have a very significant demand in the next 10 to 15 years, but then tail off. Which gases, hydrogen being a key one, will be needed in the longer term future, and what sort of materials are our starting points that are going to provide those. Many of you may be aware that there's significant import of overseas material to the UK at the moment, and one of our focuses is on looking at how much the UK can produce indigenously and what the issues around that are. So those come through in the case studies, and there was a report published on our case studies just last month, I think. It is available on the website, but what I'm going to do is pull out 2 or 3 in particular to show you how they're relevant, I think, to the Catalysis community. Much of what's down at the bottom of this slide is really, really important to us, but I'm not going to talk too much about it because it effectively replicates what Chris has just said. We do the same thing. We do the early career thing, we do the policy thing, we do the wider analysis, environmental work, and so on. So let's move on and take a close look at the topic group that Chris co-leads, which I think is possibly the most relevant one to many of you. And this, like I said, Imperial, Aston, Manchester, and Katie Chong is our topic group representative on this, and it's actually she who will be chairing the session this afternoon. So she's one of our earlier career lecturers who actually has a management role within the hub, and she's particularly interested in supporting early career researchers. So please take the opportunity to connect with her this afternoon and beyond. So the sort of things that are done in the topic group on pretreatment and conversion are, we're looking at biofuels from waste wood material, heavily contaminated feedstock, and here it's about what contaminants are likely to arise and what impact those will have on conversion efficiency. Will they poison the catalyst that we have downstream if we're trying to convert through to aviation fuels and so on? We are looking at the economics around that. We're looking at the circular economy approach. And so we're not just looking at energy here. We're looking at conversion through to renewable materials, bioplastics, and other products. I'll move on within that. I thought I would show some real science, even though I'm only doing an introductory presentation today. So this is the most, I could say this is my most recent publication, but it was actually done by my excellent PhD student up in Manchester, Samira Garcia Frietes. And this is work that she has done around gasification of coffee wood in Colombia. Now, that may sound a very long way from where many of you sit, but I do urge you to have a look at this paper, and I've given the reference on the bottom, because what it's actually all about is looking at the whole system and its overall sustainability. And it doesn't take a genius to work out that the green bits are things that are good, the orange bits are less good. And what we've done here is we've looked at different ways of producing certain energy vectors. And then we've looked at how those different vectors might fit into the energy system and what the net impact of that is. And I think the take-home message here for people interested in catalytic conversion is that it's all very well to focus in on a narrow area, and we really need you to be trying to optimize conversion efficiencies, improve them, get them up to minimum levels. But actually, we need to step back as well and look at the quality of the product that you're dealing with and what your starting point is. Because if that starting point changes and you end up with a different composition of feedstock, how does your system respond? If there are impurities in there you didn't expect, what does that do to our conversion efficiency and our economics? If you try to produce a different product, if we are looking at a different gas with a different quality requirement, how does that affect the whole system? And what Samira showed quite neatly here was that there was a great sensitivity to the technology and its performance, but also to what you were doing with it. and how it sat within the local use system, what vector you were displacing, and what feedstocks you were looking at. So I think that's just an example of how I feel there is a need for better communication between people who are focused on conversion and people who are trying to do wider systems work. I think it's really important that we understand what the limits and constraints around our respective areas are, because it's only by bringing those together that you really get processes that are environmentally sustainable. Okay, I said I'd talk briefly about the case studies in SuperGen. I'm not going to go into great detail. There is a recently published report on this, but I've just picked out the two that explicitly mention catalysis. So we have a case study looking at biorefineries, where we have ionic liquids, fermentation and catalysis to look at production of a range of liquid fuels and chemicals. Now, the sort of conversation we would be interested in having with the catalysis community over this are which liquid fuels, which chemicals, what are the benchmarks over quality and what can be achieved. So what are the feasible process routes to different options within that? I'll quickly show you another one. This one is much more specific in that the vector has already been defined. It's hydrogen, which many people are focusing on for 2050 and net zero ambitions. So we're looking at catalytic conversion of food waste on this one through to hydrogen. But again, it's about the variables in the feedstock. How will the catalytic processes respond to different contaminants, different compositions, under different conditions? Okay, I just wanted to finish with some opportunities around SuperGen. I think one of the key things for many of you working on the underpinning science around catalysis is that SuperGen provides an outlet for your data. If you can tell us conversion efficiencies, and you can tell us conditions, and you can tell us what's going on, then we can incorporate that into these system studies and provide really useful outputs as a result of that, get your work noticed, gets it cited, and incorporates it into the wider system, which we can then use to bring to the attention of the wider energy systems community. Miriam and Andrew Welfel up in Manchester are both working on those case studies. So if you think that you have interesting stuff that could feed into those, please look at the case studies report on the website and get in touch with them. We have a network for early career researchers, and I've put down the names here of the 3 chairs at the moment. Scott Banks in Aston, Samira up in Manchester, and Abdul in, I'm going to forget this, I think Abdul's in Nottingham, I hope I've got that right. And for that, you can sign up on our website. Lots of opportunities there for training and very critically for networking around the ECR base. We have funding opportunities. If you register on our website, you'll get told about those, and we have 2 coming up in September. One focused on our flexible funding. Flexible funding you can apply for at any point in time. So we take ideas. If you're maybe working with an industrialist and need £20,000 to test out a particular idea, you can apply at any point, and we review those within 3 months. But we also have more strategic ones, and I can see that I managed to not finish my bullet point 3, which was going to give the 3 topics for our September flex funding. But what I'm going to say is we're having webinars in September around our key topics before we launch the flex funding on that, and that will be for projects of up to 100,000. We also are going to be launching a fellowship call that Katie will be helping extensively with, and that will close before the end of the year. It's a mini fellowship, so it's going to provide about 1 year of funding for an early career researcher to do something independent. It's a really golden opportunity if you are looking to progress your academic career and are at an early stage at the moment. There are opportunities around impact. Andrew Welfel is currently developing a smartphone app for bioenergy. He would love people who have knowledge of different conversion processes or just like playing around with apps and can see potential within that to get in touch with him. And we have lots of policy connections and policy briefings. So many of our researchers are regularly presenting to different government departments. Training opportunities, we did a training session recently on how to produce videos to share on social media. We're trying to adapt our training program now to fit with the world that we find ourselves in. So if you have any suggestions around that, please do get in touch with Katie Chong. And my final comment is a save the date request. We will be having our annual assembly this year on the 16th and 17th November. I'm still hoping that we might physically get together somewhere, but if not, we will run this as virtual. My recollection of the timetable is that the early career meeting was going to be on the morning of the 16th, and then we would run from midday 16th to midday 17th with the main technical sessions. So do have a look at the website. I've put my contact email in there in case you want any further information. And Emma Wild, who is much more likely to respond quickly, is the project manager if you have any other queries. Thank you. And I'm just going to look and see if there's anything within the chat. When is the deadline for the fellowship application and when is it due to start? It isn't an annual call. No, we hope to have 2 calls though. So the first call will be this September and the deadline is going to be before Christmas. Probably, I would have guessed maybe 15th, 20th of December, but we have yet to set that.
Okay, any other questions for Patricia? Okay, if not, then thanks very much, Patricia, and we'll start the main meeting. So the first speaker is Mike Bowker. He's going to talk about liquid sunshine, apt from where he's sitting at the minute. And it's 15 minutes as a talk and 5 minutes questions. So,
well, apologies, yes, for the background, but I'm on holiday at the moment. Unfortunately, I'm not on holiday on that beautiful island, but I'm on holiday on a different beautiful island, which is called Britain. So this is to hide what's going on in the background in this house. So there we are. Right, let me try and share the screen then. Where is it? Right, can you see the cursor? Yeah, good. Okay. This is where we work at the Cardiff part of the Catalysis hub. This is the main building here, and chemistry houses most of this building, and so does the Vice Chancellor. But we also do a major part of our work, particularly characterizing catalysts, but we also do synthesize them and run them at Diamond Light Source. Those of you with a good eye will notice this is quite an old picture of the Diamond Light Source. We were the research centre at Harwell, RCAH, which is actually there, but not in this picture because it wasn't built then. And at the moment, we're building a new centre for catalysis at the Cardiff Catalysis Institute at Cardiff, and this is the building in construction. We share it with one other group, and we hope to be moving in there in 2022. So it's important to step back a little bit, I think, before we go on, to remind ourselves what the hell it is we're talking about. And what we're talking about is ameliorating climate change. This is a slightly alarming view of temperature change from 1850 to now. And what you can see from that, it'll show again in a minute, I think. This is from Ed Hawkins at Reading University, roughly zero in 1850, increasing during the Victorian era, then sort of roughly steady until recent times when there's been a huge increase in global temperatures. And this was, I think this was a couple of years, 2016, this was, so it would go up a bit more. Actually, maybe not at the moment, maybe it's gone down a little bit at the moment with reduced industrial activity and so forth. So we have, as a result of climate change, we have a new energy landscape, obviously, but we should remind ourselves that almost all our energy is solar energy, made by plants converting CO2 and water into carbohydrates of one kind or another. And the main role of mammals and animal life on the planet is to reverse that equation and produce CO2. And we, as a human race, of course, have got various other clever ways of increasing CO2 in the atmosphere. So we need new thinking. Or is it new thinking? Let's look at this. This is from a novel from a little while ago. And what, some of you might recognize it already. And what will they burn instead of coal? Asked Pencroft. Water, replied Harding, will decompose with elements. Water will be the coal of the future. So that was written in 1874 by in his novel Mysterious. So, as we always say, there's nothing that new in science except the application of science. Can you hear me okay? Everybody hear me okay? Good. I just had a little message saying your sound is unstable. So perhaps I'm speaking too fast. I'll try and speak a little more slowly. So this is from the SuperGen Bioenergy Hub website and shows the various feedstocks and conversions and uses it is the ambition to fulfill at some time in the future. And work obviously on all of these areas is going on at the moment. As far as the Catalyst Hub and my involvement with the Catalyst Hub in particular is concerned, we've mainly been focusing on converting CO2 from this so-called no capture route into liquid fuels and gaseous fuels. And when it's methanol, a lot of what I'm going to be talking about is methanol production. Of course, that's a feedstock chemical for the chemical industry too, so we can make a lot of base chemicals from that. But as well as that, we're involved in various catalytic ways, low temperature generally, catalytic ways of converting biomolecules using catalysis into upgrading them into higher value products. And so there's a number of specific projects at Cardiff relating to this. I'm not going to go through exactly what they are, but in all of them, what we really do is make catalysts, make sure we have state-of-the-art technology to characterize them at the nanoscale and atomic scale. And we use a lot of novel methods of catalyst synthesis too, innovative methods of catalyst synthesis. And of course, at the end of the day, these have to go into a reactor, because without that reactor information, we don't really know whether these things are any good or not. And all of that is a feedback loop to try and make better catalysts. So we're interested in making energy from wind, well, from solar energy, of which wind is a manifestation, but also via photovoltaic. And at the moment, the main route to energy is through hydrogen production, as we just heard, using electrolysis. That produces hydrogen and oxygen. The oxygen at the moment is a valuable product because it's medical grade oxygen. But using that hydrogen, we can then produce other fuels. Obviously, hydrogen is very light, so we have to either compress it at high pressure or cool it, make cryogenic hydrogen to densify the fuel in volume terms. Another way is to make liquid fuels like methanol, or gaseous fuels like methane, which we can feed straight into a grid, or ammonia production is another route to storage of this variable energy that we can use. So the idea is, one idea is we make these liquid fuels or gaseous fuels or solid fuels at times when energy production from renewable energy is high, for use at times when that production is low for one reason or another. I'll talk about that route later. So we're interested in producing a variety of molecules from CO2 and hydrogen, hydrogenation of CO2 to methanol, dehydration to dimethyl ether, and the production of fuels from there, a so-called methanol to gasoline type process, or using the reverse water gas shift with CO2 and hydrogen to make CO, and then using that CO through Fischer-Tropsch type catalysis to make olefins and hydrocarbons for fuels. So let's remind ourselves where we're at with this. Methanol synthesis is currently carried out in typically plants producing 2 million tons per year in individual plants of methanol, and most use the ICI low temperature process, which was invented in the 60s, which is basically copper, zinc, and alumina. Copper, zinc oxide, and alumina catalysts with various additives. They will only work well in a sulfur-free stream, so there's a trapping system in front of the catalyst that does this reaction to get rid of sulfur from the gas, which is has sulfur stenching agents in it. So that is actually the catalyst down there that's used. It's a pelleted form of this copper, zinc oxide, alumina. And one of the crucial things about that is it's strong, very strong. Important thing for catalysts in a big plant, because there'll be typically 100 tons of that stuff in there. I don't want you to look at the details of this, just that bit, bottom right. Methanol synthesis works in that way at the present time. It's a mix of CO, CO2 and hydrogen to make methanol, but actually the oven is carbon monoxide conversion to methanol in a dry process. And that uses fossil fuel. It's fossil fuel, hydrogen from methane, usually as the source. What we want to do is a different reaction. We want to hydrogenate CO2, and the big difference there is that we get a mole of water for every mole of methanol we make. And there are some problems with catalyst stability in that kind of situation. So at Cardiff, we're busy looking for novel materials for methanol synthesis, ones that may be more stable, more water tolerant over long periods of time. And a particular focus of ours of late has been alloy materials of various kinds. So I'll just talk about one at this point. We were going to make palladium with various other metals as alloys, just as in the first instance, to see what we do with that for this reaction. And why palladium? The reason we use palladium in the first case is that it doesn't produce much methane from CO2 and hydrogen, at least so we thought, and actually it doesn't. But what it does do is it does very efficiently the reverse water gas shift reaction. So if you look at the curve on the left, this shows that the catalyst produces mainly CO at low temperatures, and then may make some methanol at higher temperatures. It converts to methanol at higher temperatures. And the reason for this is the catalyst reduces in this hydrogen CO2 mix, and it reduces to make a palladium zinc alloy, which was a surprise to us at the time when we did this work originally. It came as a big shock. And of course, what that means is if we pre-reduce the catalyst in the right-hand panel, we make high selectivity to methanol at low temperatures, at reaction temperatures, which then goes down due to equilibrium considerations, and CO comes up at high temperature. So palladium zinc as an alloy is a good catalyst for methanol synthesis. And we've shown in other work that it's actually more stable than copper-based catalysts. Of course, this is an academic study. We may well not want to use palladium in such catalysts. It's not an earth-abundant metal. And we can go on from there to change the properties of that material by including an acid function. And if we include an acid function, obviously we can then have the possibility of dehydrating the methanol to dimethyl ether, so we can get high yields of dimethyl ether when we create in the correct way a mix of palladium, zinc alloy, zinc oxide, and an acid function, in this case ZSM5, but we've used various other ones. So the right-hand panel here shows a peak in dimethyl ether production at around 280 degrees centigrade or so. If we go on even higher in temperature with such a catalyst, we can start to make hydrocarbons too, and do a kind of methanol, or in this case, direct, this is all in one pot, direct CO2 to gasoline type products. In the Europe that we evolved, the idea was to go a step further and actually construct a complete plant. So this is a plan of the plant. It's a 500-ton-a-year plant. And this was constructed at a coal-fired power station in Niederhausen in Germany and had a number of players involved. First of all, we need the coal-fired power station to produce the CO2. We then need to purify that CO2, react the CO2 with electrolytic hydrogen. Hydrogenics here is a major producer of electrolyzers. Those go into methanol synthesis. CRI Iceland is a producer of small plants and has a plant in Iceland, and it produced a plant to produce this amount of methanol in this project. So this is the coal-fired power station. This is the second biggest cooling tower in the world. And there's a little space here for our methanol plant next to it. And here is the methanol plant, nearly finished. It wasn't actually quite finished at this point. This is the electrolyzer. That's the methanol synthesis plant. and that finished its work and produced methanol at the rate desired, and was then shut down at the end of the project in July or so last year, and was then shipped to a new project in Sweden. In terms of interaction with the SuperGen facility, then of course, there are a lot of units around the world now in farms that are biogas units, in which, for instance, maize, this is actually a farm near where I live in Oxfordshire, and this converts slurry waste, mainly pig slurry waste, or maize, or both, in fermenters to produce methane. And it produces about 20 tons of methane per day. The problem with that is it also produces about 20 tons of CO2 per day. Not 20 tons, but the stoichiometric relationship of CO2. And they really would like to use that CO2 instead of venting it to atmosphere. They can, of course, sell it to the drinks market, which is where a lot of CO2, well, not a lot, some CO2 goes to. CO2 out of ammonia plants goes to the drinks market. But it would be better, we could imagine utilizing that CO2 in a small methanol plant of the size that I've just talked about to make methanol as a higher added value product to sell. There's also lots of other catalysis related to the BioSuperGen platform. Since I've just listed 3 here, this is off our Cardiff Catalysis Institute website. Glycolysis with glucose, cellulose conversion to gamma valorolactone, that's a bit of a mouthful, and aqueous phase reforming of carbohydrate mass to produce hydrogen, biohydrogen, for instance. So there's a lot of work going on in that direction too. I showed this slide earlier. One thing I didn't mention was this, of course, we have to go through electrolysis to produce hydrogen. It would be rather nice if we could do it directly from sunlight by splitting water using photons from the sun. Of course, that's very difficult. So here's an ideal. Hydrogen and oxygen from water, photocatalytic water splitting. That's endergonic by about 280 kilojoules per mole uphill. So it's extremely difficult. There are those around the world who claim they can do it at the moment. But what many people do is use a sacrificial agent with the water to extract the oxygen from it and to overall produce hydrogen and CO2. And you can see from a paper we published, and Chris was involved with this. This was mainly Queen's University, Belfast and Cardiff. We could convert cellulose to hydrogen using photocatalysis directly. This is in a very small pot reactor with a very small amount of material. But even grass, this was grass, I think, from your garden, wasn't it, Chris? Fescue grass.
My garden.
Which had very little processing. I think we dried it a little bit and we pulverized it a small amount. So grass was put in the reactor, and lo and behold, that can produce hydrogen too. You have to be careful sometimes doing these kinds of reactions because you also get lots of reactions in the media. If you look at the middle one here, this is from the Oregon Telegraph, believe it or not. Grass power coming soon to a home near you. Well, how soon is a question, another matter, but anyway. So to summarize, this new energy regime we have for the next 20 years gives us new opportunities, lots of them. The biorefinery idea to produce fuels and chemicals will require lots of catalytic processes, just as a petrochemical refinery does too. And as a result of all that, as we heard before, obviously there's a clear synergy between the SuperGen Bioenergy Hub and the Catalyst Hub. So at that point, I'll finish, and I guess I should escape from that. Now, how do I get back to questions?
I can answer the question. So the first question, Mike, is from Michelle Lynch. Do you know how the Carbon Recycling International methanol synthesis catalyst copes with water?
Yes, that's a very good question. And what I'd like to know too, within that project, it was quite tricky because they're a very secretive organization. So they brought their bit of this whole construction in a black box, and only their people worked on it. So the answer to that is I don't know. That plant was only meant to operate for a very short period. It operated for about 2 months. It was only meant to operate for 2 months in this kind of project, because most of the work was constructing the elements and then putting it together finally at the coal-fired power station. So I think the answer to your question is, I don't know.
Okay, thank you.
It worked for the time. It worked for 2 months very well.
Right. Keith Simons, you mentioned surplus electricity. What percentage of electricity is surplus? Your electrolyzer is expensive. If it is not being used for a high percentage of the time, there's low capex utilization, which will impact on your economics.
Yes, so part of that project was at the University of Genoa and it involved economic analysis of the whole process. And you're right, you've got to import electricity at some parts of the time and export it at other times, which relates to this high productivity, low productivity from renewables. So costing was done of that, and basically it's 500 ton a year plant is non-economic. Obviously, it's just too small scale. But once you get to 50,000 tons a year, using wind power, not photovoltaic, it became profitable within the parameters that were in there. And parameters are things like the oxygen selling price, the price of methanol, and so on, price of electrolyzers, which is a big cost. And electrolyzers is a big cost in a plant like that. I think what we should say is this is part of a toolbox. It's just one part of a small part of the toolbox of technologies that we're developing for application in the future.
Thanks. Can you please give an analogy between biofuels versus electrical? Well, that's from Deva Dutta. Deva Dutta, could you clarify your question? You're on mute, so.
What I think the question is, oh, sorry, go on.
Yeah, Mike, this is Devadatta. Actually, in terms of transportation, while we see in the future, so people are coming with electric vehicles. So how far the liquid biofuel will be like sustainable for the future?
It's a very good point. I mean, don't forget, if we're making something like methanol, that's a platform chemical for the chemicals industry. So it's part of a possible green future for chemicals production rather than using an oil base for chemicals production. But yes, the fuel, don't forget, can be reused to produce energy at times when energy production is low from renewables. So yeah, I don't envisage that in the long-term future we'll have fuel-driven cars, except for hydrogen-powered cars and electric cars.
Okay.
You're right. Yeah.
Thanks. Last question. You did not quite say what the alternative to palladium is, given it is a rare, expensive metal.
Yes. Okay. So, I mean, that was just one example of something that I found interesting and came as a surprise when we did it. We're all also working on, I said, of course, you wouldn't choose nickel as a starting point, which obviously is much cheaper than palladium. You wouldn't choose that because a methanator, if you want to make methanol. But of course, there may be ways of manipulating even nickel to make it a methanol catalyst. So we're working in that kind of direction. We're also working on new types of copper catalysts that may be more stable, more active, and work better in that kind of environment. One of those was part of that project that I mentioned. And at the moment, I can't really say much more about it, but we've made some higher area copper catalysts by a particular route, which may be, and they've been tested in industry, which seem to be more stable than the normal industrial catalyst. And we'll take that further in the future.
Okay. There's lots of questions. I think, oh, and lots of answers as well. I think we'll leave it there, Mike, and let you carry on offline if you want to. Is that all right?
That's
okay.
Fine. Thanks, Chris.
Thank you very much indeed, Mike.
Okay, everybody. Bye then.
Oh, Pip, you have to sit down again. He got up when I started clapping. Right, okie doke. Right, let's move on. We're a little bit behind time, so we need to move on a bit. So the next is Mohammed Naveed. He's going to talk about an overview of resources and land use within SuperGen.
All right, so I hope everyone can see my presentation. My name is Naveed and I'm from Aberystwyth University. I'm special modeler here. So I'm presenting here on behalf of topic group resources to give an overview of resources and land use within SuperGen. So there are some research questions like how can we grow biomass on low-grade marginal lands, and how can we make more crops which are more resilient to extreme weather events, especially under changing climate, and also to deliver ecosystem services. And there's another question: how can we have energy crops which can we can reduce the cost of production to have our bioenergy crop establishment, and to how to increase our sustainable increase in our crop yield, and how we can map our waste streams. So another question is how can biomass be matched and to the end user requirement. And there is a fourth question on how can we maximize the environmental benefits of energy crops and the waste management, and how we can minimize the negative impacts of these. So we choose, sorry, I did not make a full screen of my presentation. So we choose the second generation crops as compared to the first generation because of the higher energy ratio and also the low input requirement per unit biomass of these crops. And these second generation crops have the ability to grow on low quality marginal lands, and also they have the higher land use efficiency and wider ecosystem services. So using of these crops along with the organic waste that can be helpful to have energy production from it. So there are various sources of sources for these biomass and for the energy production that start from the plants, timber, to agriculture, food waste, or even the sewage. So miscanthus is one of the example, which is a really good type of environment friendly crop, which require low input to give higher biomass from it. And miscanthus along with other willows is another example of the perennial crop in UK, and it's been grown for a long time. And typical lifetime for miscanthus is around 15 years. That can produce on an average of 13 tons of dry matter per hectare per year. And willow is also another good example for these bioenergy crops that can grow from 25 to 30 years of its lifetime. And it can give around 7 to 10 tons of dry matter per hectare per year. So using these biomass and waste feedstocks for energy and transport fuels, we can generate electricity and gas generation from it. And there are other agricultural residues from our garden that are maybe from the field, like the straws from the cereal crops, or the forest residues that can be used for the feedstock. And these feedstock can be piled and built close to the source, and it can be transferred to the gasification or the other facilities. So here's a complete cycle of starting from the crop to its and transportation of these feedstock to the source for to different processing units where it can be used to generate electricity. So there are various tasks which were decided. First one was assessment of marginal land. That involves which type of new crops, new varieties, or alternative crops which can grow. We can grow, and we can utilize, maximum use of these marginal land for our energy crops. and to use various type of modeling techniques to identify the areas which are best for specific crops. Might be miscanthus, willow, red clover, white clover, or other type of energy crops. Another task is to understand the soil and climate heterogeneity of UK. That involves using the climatic dataset from UK Met Office to generate the maps and the climatic zones of various parts of UK, which type of climate that exists in these parts, and what type of soil, various type of soil or soil variability exists in this area, in this region. So the third task is modeling of ecosystem impacts. There are various special models that can be used to identify the impact of different cropping system on growing of on ecosystem that involves if there is how we can have the flood mitigation, soil quality, carbon sequestration, and to keep the biodiversity in that area. And the fourth task is the opportunity maps that involves in which part of the UK we can get the maximum yield if we do some sort of management practices, or if there are some area which is low grade land, which type of crops we can grow in that area. It's all about land suitability. So when we talk about the marginal land, there is different definitions about the marginal land. Even it's really broad in concept, but a generalized understanding and knowledge of marginal land is limited, and it is very diverse because it depends on it varies on across the country's local condition and the researchers or the organization which are actually studying that issue. So there are some qualities, same qualities used to classify a site which can be marginal for one type of purpose, but it can be productive for other type of purposes. For example, if there is the land can be marginal for a specific type of industrial crops, but it can be productive for miscanthus or other type of energy crops. So there are various constraints or challenges for our land use that involves climate constraints, geophysical constraints, socio-ecological challenges, and economic challenges. These all involve to define a land as a marginal land or a productive land. So I'm working on a spatial mapping of current resources availability and predictive maps for future availability that involves using the climate, soil, and other land attributes data to start to parameterize my model, which is a miscan-for model. Is being developed by Aberdeen University scientists, environment scientists, and I'm working with them in collaboration from Aberystwyth University to generate maps, predictively maps for various bioenergy crops, and also to identify the maximum potential of bioenergy crops in various parts of UK. So using all these spatial data set, I'm developing maps for the whole UK to have some potential areas for various type of bioenergy crops. So here's one example of the crop map which I have developed before, which is only for the Wales, but I'm using it the same methodology for the whole UK to generate predictive miscanthus and other crops maps for the whole UK, which part can give us the maximum potential of those crops. So another is organic waste prediction that involves using various organic waste sources. Start from the waste collection from the households, from the garden and other parts, and to generate electric and to transportation from those waste from the field or from the households to the plant for its processing. So my purpose is to generate the current what's the gap between the demand and the total availability of these organic waste, and how we can increase, how we can predict for the future that how much we can get these organic waste from various parts of UK using the modeling technique. So this is all about very brief introduction about my work. So let me see if there is some question. Chris, can you help me?
I don't have any questions for you at the moment. Perhaps I could ask a question in terms of the use of biomass from the UK compared with other countries. What are the major things that we need to be considering in terms of the diversity of that biomass?
Right. Yeah, everything in the waste, if we want to increase the efficiency of our waste, to use each composition of these waste to be used for electricity or other purposes, even to recycle all these wastes. So we really need to I'm comparing actually it with the other parts of, even in the Europe and also in United States, that what type of things they are using it. So all type of waste that start from the garden, from the food waste, which is being collected weekly or whatever in different parts, and also the non-recyclable food, the waste, and the waste which is from the forest, the waste which is from the sewage, and also we are also going to include some waste from the contaminated water. And especially the wastewater, which is like maybe the grease or fats from the water, to use it for our energy production.
I suppose my question was more along the lines of, if you've got waste biomass, like you said about forestry waste and so on, the conditions in which it's grown can make a big difference to, say, the lignin, hemicellulose and cellulose content of those. And therefore that will then determine what you can do with that material and how much energy you can get from it. Now, what sort of variation do you get comparing, say, Australia or Asia or the UK, for example, in forestry waste for the same type of wood?
So still, I'm working on it. So I'm not sure about what is the main difference between the different waste sources, to compare that which is more efficient as compared to the other one. So hopefully I will get more information to share on.
Okay, thank you. We have a question from John Irvin. For miscanthus, are you looking at centralized utilization? If so, are there energy costs in transportation?
What is centralized utilization? Is maybe
taking all of the biomass into one place and then doing it, rather than doing it locally?
It's better to be centralized, but the processing can be maybe in different parts. The processing can be in different locations, which is more closer to that source. There will be more cost of transportation if it is being centralized, but it's better to have different parts in which is more closer to, because when I talk about the cost of production, so that also involves the cost of production. Another side of the cost of production, then when the transportation cost is more, then there will be no benefit from it to have either to have use of these miscanthus. But if the crop is growing in specific part and its transportation cost is lower, then we can have higher, more use of that area as compared to the centralized system.
Okay, thank you. Yeah. So how would you factor the seasonality how would the factor of seasonality, sorry, be considered for different types of feedstocks?
Yeah, this is also very important. The availability of these feedstocks is in different seasonality is also important to patch of the continuous availability of these feedstock. So we are considering various factors which is being involved in it, because the availability of from the crop side, for example, if I take the the example of miscanthus, its availability its maturity and its availability is in the end of the season. But the other resources which can be like for the other crops, which most of the crops which are available and harvest season is in October, before October. So it's very important to patch and make a proper timeline for these availability of feedstock. Yeah.
There is a comment about whether you're going to use diesel to transport the miscanthus.
Sorry, what?
When you're transporting the miscanthus, would you use diesel, as in diesel trucks? Or
yes.
Are you looking at other things?
There is a possibility of using the other things for a big area, not too big, but for a big area. If the transportation facility is centralized, not for the big, like for example, if I talk about one county, if I consider like that small unit or small zone, then if I consider it like centralized, then the cost will be reduced. But if it is for the big area, then there will be more cost for it.
Okay.
And, yeah.
Right. Brilliant. Thank you very much indeed. I don't think there are any other questions. So, Naveed, thank you very much indeed. So, the next talk is by Arthur Garforth, who I know very well. And he is going to talk about, if I can, too many screens. The problem with Zoom is that I have, oh dear, my screens have gone black now, which is not good. Oh dear, right, okay. I would like to tell you what Arthur's going to talk about, but my computer's decided to have a bit of a wobble. There we go. The circular economy and the upscaling of polymer waste to chemicals. Arthur, take it away.
Thanks, Chris. Since my computer just crashed, here's fingers crossed. I hope everyone can see the screen, in which case I will proceed. So thank you very much, Chris. So working within the Catalysis Hub, my area of expertise, I suppose, is heterogeneous catalysis and how they're applied to a difficult problem waste. I'm just trying to see if my screen will go to full screen, which is delayed. 1 second. Yes. Okay. So the circular economy and the upscaling of polymer waste to chemicals. Now I'm picking up on a lot of things that Patricia has mentioned earlier about we can convert things and we've got to consider the wider system. So I hope I'm going to bring you a little bit of that in this talk. Also, of course, you can't get more varied than plastic waste. So your feedstock sensitivity, your impurities, and the quality and consistency of your products are all key problems, challenges, reasons to say, no, it's not possible, and so on and so forth. So without further ado, here's a bit of background information that not necessarily everybody is aware of. So there's a report on global plastic flows published in October 2019 on moving towards circularity. Just so you know that not everything is circle. You can also draw this in a linear plot, but let's just take circularity from production all the way through to consumption, waste collection, recycling, and recycling. Notice how the numbers suddenly drop off as you move from the production to the recycling. So if you go backwards on that loop, you can see that that's quite a significant, it's less than a sixth of the amount being actually recycled. The waste collection is the bit that causes all the fuss. The consumption, we don't care about. We eat things, we have packaging that covers it. the recent refusal to use plastics has been knocked on the head somewhat by COVID-19, where now everybody, I believe Sainsbury's, Tesco's, Morrison's and others, thrown away tons and tons of vegetables that you can pick and put in a bag. As consumers, of course, go to something that is protected in a plastic carrier or plastic container, which of course these are very complicated. Now the key thing that caused the problem and the public outcry, and still causes the public outcry, and now contains COVID PPE, is what we see in the streets and what we see in city centres, and it's all that improper disposal and leakage to the system. So if we look at the ways that these things are normally recycled or managed, we dump quite a lot into a landfill, we burn a bit for energy recovery, but we have a lot of plastic that gets inappropriately disposed of, even more fly tipping now than ever. On a local note, I see Sainsbury's has locked off all its recycling. In the area where I go shopping, the local population has decided to pull away the screens and fill up the bins, even though they're not being taken away. So there's a bit of education that needs doing around society, as well as an education around how we might do something with them. So it's not so unseen anymore. And of course, that has caused finally a wake-up call for the plastic manufacturers themselves, and Plastics Europe that you see at the bottom there are a group that represent all the European plastic polymer manufacturers, suppliers, recyclers, and so on. And the organisations in the UK, British Plastics Federation, Recoup, have many initiatives, and there is an annual conference, or yes, there is an annual conference which deals with recycling, which is held in Wembley, and Identiplast was the last one. These are all available publicly, these documents, so please look at the websites of those people. So what happened is the Plastics Europe did a bit of research, and they found that, not too surprisingly, when we go swimming, my other half is very nervous around jellyfish. So when you go about a meter into the sea, you very quickly see plastic bags, and there's a scream because we think it's a jellyfish. It isn't, but this is the sort of stuff that Plastics Europe surveyed all the European beaches, and these are things that appear on the beach. Some believe it or believe it not, but mostly that's one of the beach. So that's our feed source, possibly. But another feed source, of course, is one that may be less known to you all, which is within the NHS. The NHS has a horrendous carbon footprint and has been trying to reduce it for a few years, not just for a couple of years. And they identified within their waste stream, top 15 equals about 69% of the products. And just so I don't expect you to take all that in, but just so that you have a look and can see the sort of things, here to protective clothing, examination gloves, and we come further down and we see things like syringes, needles, clinical waste, medical packs, and so on and so forth. But that is 72,000 tons, or was in 2016, I think this is from, and you can see that if it was only 69%, then there's at least another 30,000 tons of waste which contains plastic in their waste stream. And I don't think you'll disagree that that is a very varied waste stream indeed. So what have I been, where's my area of interest? Since 1994, as a young postdoc, I was interested in recycling plastic waste, and feedstock chemical recycling, as it's known, or chemical upscaling, consists of several routes, 3 of which are there. Thermal cracking is one that receives a lot of attention because lots of people set up in garages and heat plastics and turn them into biodiesel, show you a lovely murky black flowing liquid, and then they put it in a nice BMW diesel engine. I don't think they do. Others can correct me. Catalytic cracking and hydrocracking are the 2 areas where I've been involved in. Catalytic cracking is fluidized bed catalysis, and hydrocracking is both are standard petrochemical plants. I thought that would be interesting. I know we all use plastics. I know we all see plastics every day, but they are ubiquitous, and there is just some of the applications that you will have in your household and in your daily lives, just to give you an idea of the scale of the issue. So when I started this work, it was obvious that you had to focus in on municipal waste to me. This, of course, was obvious back in 1993, 20 so years later, and it's obvious to everybody now that we need to do something with these problems, but it's a very difficult problem and very mixed waste stream. And even on that little graphic there, you can see the different colors, which automatically give you additives which give you poisons. But if you look at that, polyolefins, PVC and PET make up about 73%. Industrial processes that might look at horrible feedstocks or feedstocks that are viscous would be fluid catalytic cracking, which is one of the interests I had until about the early 2003. And basically, you're putting a catalyst, if I may use my mouse, putting a catalyst in jointly with a feed and fluidizing it, cracking it, and recycling the catalyst and adding more feed. They're typically zeolite catalysts mixed in an amorphous silica alumina with clay binder, and the scale of the plant you can see there. The product stream, I'm just showing you this graph. I don't want to bamboozle you with information, but the left-hand side is weight percent. The x-axis is showing you, in this case, light olefins between propene and hexene and saturated equivalent on this side. And you can see when you play around with the zeolite or with the amorphous silica alumina, which is the gray one, you change the product stream. The key thing here is olefins are a key product, and there's a key shortage of these materials in the world because they're needed for polyolefins. And if you can turn plastics back into some olefins, then you're automatically going to generate a feedstock that's useful. The other way to look at it is that whole picture there equals some light LPG with olefins and some light naphtha. Naphtha can be steam cracked, as SABIC and other companies will tell you, with steam crackers, and turned into olefins, which then get turned back into polyolefins, which add to the circularity. So by doing this, you're keeping those molecules in play. However, it's a highly endothermic process. It's also prone to, in a technical term, gunking up. It's highly viscous material. Fluidized bed catalysis is set up around gasifying gas oil or slightly heavier vacuum gas oil, and therefore it has problems. Hydrocracking, however, is energy neutral, slightly positive to quite exothermic, depending on how you set it up. It also likes to deal with other contaminants such as sulfur and so on. And the typical conditions from the plant is shown there on the right-hand side. But the typical conditions of hydrogen pressure being at 80 to 200 bar immediately puts researchers off in the lab because of safety. But the temperatures are quite moderate, 300 to 450 degrees C. Now, I will say I owe a great deal of debt in this presentation to a company that has already had a mention by Mike, which is ICI, because I was able to go into the Runcorn high temperature and high pressure labs and carry out some work and do real catalysis at high pressure in their safe environment. So I started this work back in 2003 and was able to carry it out in their labs for 3 or 4 years as a young lecturer, working 1 day a week in their facilities. So anyway, so here's something you'll recognize. I normally have a series of slides here, but I thought for brevity and also to show you that we've moved on from just using pure feeds, so polyethylene on its own, or high-density polyethylene and so on its own, we can do that. That's no problem. The issue now is to try and use real feeds. And here are just a graphic to tell you the real feeds that we're using. So you might be well familiar with the centrifuge tubes if you're a laboratory chemist, biochemist, and the wash bottles, which you see here. As general human beings, we're very familiar with water drinking cups and with plastic milk bottles that we buy. Note the immediate complexity of the system. You have labels, they've got inks, you've got colors. Note that the manufacturers are responding to the recycling, as you may know from experience yourself, that water bottles are not as thick as they used to be. And the color on the top of a milk bottle is 75% reduced now, so you can actually see, it's not see through, but you can certainly see light through it as manufacturers respond to trying to make things more recyclable or save dyes, save pigments, and make the environment a better place. We've done some work on these, and the conditions are shown in the table below. And 330 degrees C, roughly 20 bar hydrogen because we're working in the lab, and agitated speed and batch time. Now, we've done work which has shown prior to the Catalysis Hub and during the Catalysis Hub funding that the actual reaction takes place a lot quicker than that. We have exotherms that show it happens in about a minute and a half. Most of the problem with using a batch system is it's an autoclave or it's a pot. For those of you who are drinking your cup of coffee, the reactor is about the size of that coffee cup. And from that, we are able to take these with a platinum containing zeolite, which is not necessarily the optimum catalyst, but is a catalyst that's used in hydrocracking processing for real on large scale. And we're able to take what you see there. So the LDPE is the wash bottles, the high density polyethylene is the milk bottle, the polypropylene is your centrifuge tube, and your polystyrene is your drinking cup. And if you can see there, I tried to show what my PhD student Abdul Rahman has shown. He's published recently in an Indengchem Res paper. Is you get LPG, plus you get naphtha and some heavier material. It's not dependent on the catalyst, of course, because the catalyst system is a mess, because it's catalyst plus a viscous polymer, which has to heat up, which is contacting with hydrogen, which isn't very soluble in the polymer melt. And the three have to get together to give you your product stream. So not the best or optimized mixing, big chem-eng problem, mass transport in this issue and mixing. However, depending on the polymer and its molecular weight and its type, you will see the product stream changing. The one that's really easy to see is the polystyrene, because polystyrene quite happily breaks down to benzene and ethyl benzene, and depending how severe your conditions are, you can tweak which way round this is. So I think that's quite interesting. We've also mixed all of those together. So those are the individuals, but we've also mixed them all together. One of the things I will draw your attention to here is the solid, as LDPE is harder to break down than the others. But this solid, of course, is partially depolymerized polymer. It's not unreacted polymer. It's just that it's harder to represent that on this slide. So what is all this about? Well, the technology and the keeping the molecules in play is basically reducing the use of crude oil or fossil fuels. That's the first thing to say. It also allows us, if we can perfect, go to the next TRL level and so on, scale it up, it allows us to produce intermediates which are instantly plugging back into a refinery. So if you look at the cracking and the conditions of 450, 550, those are standard industrial process temperatures, typically 530 an FCC unit runs at. Hydrocracking, typically between 300 to 400, more reasonably 350, I would say. And you're turning out a stream, in the top case, catalytic cracking as olefins. In the bottom case, you're producing a range of saturated, highly branched materials. Now, initially, you could think of those as fuels, or you could think of that naphtha as a source of producing more olefins and increasing the circularity. However, the advantage that hydrocracking has, and you may not have noticed that sleight of hand as I popped PVC up on the top right-hand corner, you are able to process a bit of PVC. Now, by a bit, we've done up to 10%, 15%. I don't recommend it, but as PVC is reduced in the waste stream, and as PVC is phased out in a lot of the products that we see in the supermarkets and in general packaging, obviously not double glazing at the moment, but as it is reduced, then it means that stream has less of the PVC in it. Now then, recent work that we've been doing with Chris, and I have to say a big thanks to the people working on that. I'll thank them at the end in a quick slide if I've got time. I will have time. Is to look at blister packs. Now, blister packs are the things that you and I use if we have a headache and we pop a tablet. They're designed, believe it or believe it not only just for the integrity of the product that's within it, keep it fresh, to keep it dry, to keep it usable over a long period of time, but also because they give a really satisfying pop when you pop them. I'm not kidding. And to allow that to happen, you use this material, and we thank AstraZeneca for the materials and their very positive engagement with us. And it's a mixture. So this form pack that makes the thing that you see with the tablet in consists of orientated polyamide, aluminium, PVC, the layers you can see there. Not often that we get a lovely micrograph that says exactly that, and you can see the same thing shown in this micrograph by Alex Tedstone. And from that, we end up separating it into these things with Adam Greer, who started the work off, and then Camille Oster, who was funded on the RE3 UKRI funded project in Manchester. Now, what I wanted to say here, you're not only separating it, but you're doing a one-step dehydrochlorination of the PVC as it goes black. The ionic liquids that we're using are trihexyl tetradecyl phosphonium cation with chloride is an obvious anion, but also we're moving into others. This work has just recently, very recently, been accepted in green chemistry and is available online. Of course, the next step for all of us budding scientists and engineers, and I'm a scientist, I have to say first, is that we need to think of, can we exploit this technology and take it to the next step? There is a phenomenal amount of bids and things that are going on at the moment. But if you, like me, have been involved in industry and worked and also tried to collaborate with industry, there's just recently been a small business research initiative by the, I'm trying to think of the name of it now, the Sustainable Innovation Fund, and it's the SBIR fund. So we're making an attempt to take the small batch and build a demonstrator, because until you have a demonstrator, I think as Mike alluded to before there, a company can come in and give a black box to you, and then they don't tell you what's in it, and you don't know what you can't really do much except give them a catalyst and try and be thankful that you're involved in the process, as I am. But in this case, we've just put a bid in, small bid, to build a scale up of the batch reactor. So remember your coffee cup, if you've got one in front of you, to take that coffee cup to a realistic demonstrator rig, maybe 1 meter cubed or 5 meter cubed, will cost you £2 million. So it's important to start, so we're engaged in that. So I recommend that any of you that have got strong industrial links start to, I'm sure you're already looking at these. Gary Leake here, who's at Birmingham, published this paper of which this chart was there, and I really like this definition from 1 to 10 of the things that you should do towards a circular economy. And if you start to think about where the coffee cup is, it lies between 3 and 4. And what are we really doing? We're recovering, we're potentially recycling. In the case of the ionic liquids work, we're actually getting all the components back and then possibly taking the PVC and adding it to other polyolefins which are hydrogen rich and turning them into naphtha. So we're still really at this bottom end of this scale. I'm conscious of time. I think I'm overstaying my welcome. So from an experienced person who's been around in this area and had a vested environmental interest in joining scientists against nuclear energy back in the day, to thinking about polymers and thinking of what a waste this material is. If we just incinerate it or stick it in the ground, one day they'll dig it up. Back in '93, the world has changed, courtesy of Ellen MacArthur, courtesy of David Attenborough, courtesy of several BBC programs. And we've got to start thinking how we might take established technology on a large scale and do something with it. So it's a balance between recycling and incineration. There's a very recent LCA analysis done by Bassett in association with Adisa Adepajek in Manchester, which shows that mechanical recycling and chemical recycling are equivalent in LCA terms, and that obviously waste for energy is a really bad idea. I'll move on. So longer established technology, Patricia's right, sorting collection, getting the right supply. Retail drivers, retailers are now driving for simplification, decreasing the amount of dyes, and taxpayers and funding currently is 90% we pay, but soon the extended producer responsibility will be paying. So I'd like to thank the various funding bodies over the period of this work, and just mention Alex and Camille, Adam and Alex again for the 2 grants we've shown this work, the Catalysis Hub and so on, and the various people in that. My final comment, do I feel like I'm swimming against the tide? I used to. but not anymore. Thank you very much.
Thanks, Arthur. Very good. Right. There's okay.
Oh, I need to unshare.
So the first is from Keith Simons. As the world's largest distributor of LPG, I'm interested in to see the high yields of LPG. Keith, do you think that this is a route forward for LPG more widely in the future then?
So there's a lot of interest from the refineries in co-processing. So there's very few points you can sort of inject into refinery with a renewable feedstock. You have to be very brave to inject into the hydrocracker because they do tend to be quite sensitive. But there's a lot of interest from all the refineries on how to do something with those assets which will otherwise be stranded. What we tend to find when we talk to people is that they say, well, we have thought about putting stuff into the hydrocracker, but unless you've got a customer for the LPG, and the renewable LPG is not economically viable. So that's when we tell them we're interested in the output. I've sent you an email, Arthur, we should talk.
Yeah, okay, no problem. Thank you, Keith. I totally agree. One of my major stumblers in this is that I can't stop producing LPG. So, and then we had an MBA group of students look into the feasibility of commercializing the project. And in doing that, they went, oh, did you know that LPG was really valuable? And I went, I was focused in on the other end, which was your aviation fuels and so on at the higher end, in the middle, and also, of course, trying to produce a safe and storable product as opposed to having something which is a gas, which you then have to rethink. Keith, if it helps you, I used to work in SGS and I used to nip over to Burma Castrol. And we had guys testing for the certifications on stuff on that site. So interesting. Thank you very much, Keith.
You won't know SHU Energy. You'll know us better as Calagas in the UK.
Ah, right. Okay.
Thank you, Keith. My job is to make 5 million tons of renewable LPG by 2040.
I can help you with that.
All help is gratefully received.
Okay, so you asked about yields. As you drop the conversion of the material down, that yield, the yield of gas is the first thing you get, because basically the catalysts that we're typically using to hydrocrack are zeolitic, and therefore they just chomp it and tend to produce gas. Depending on the structure of the polymer, LDP, HDP, polypropylene, you tend to get a propensity of isobutane. So it's very interesting. I'll stop there. Thank you, Chris.
Just following up on that, Arthur, if you run at low conversion, therefore you produce high selectivity to gas instead of liquid hydrocarbons. If you carry on doing that at low conversion effectively as some sort of recycle loop, do you carry on producing gas though, or do you then start to gradually form the liquid hydrocarbon?
That's a good question. So yeah, you're right. So what you would I think what you would do, I think you can't do it in isolation. Keith alluded to that in his question and part of his answer as well. You can't do it in isolation. You're going to get a product stream that you tweak to. So if you want to be better for the environment and so on, then you can drop the temperature a bit, which again, if you're in industry and you're doing a moving bed kind of thing rather than a batch kind of thing, you can tweak the conversion the way you want it. But you're right, Chris. Yes, you can't just make LPG. You've got to make LPG and something else. And as you keep adding, it's going to give you liquids. Of course, it is.
Yeah. Okay.
LPG and naphtha, actually, Chris.
Okay. Well, that might not be too bad, though. So.
No. I mean, the naphtha is of value because, of course, and this is one of the things that the key stumblers there. If you get chlorine or if you get sulfur and hydrocracking, hydrotreating can deal with that. So you'd have to have bespoke units, kind of what Keith alluded to as well. You'd have to have bespoke units. And therefore, I think you've got to watch what you're doing to the equipment that you're doing it, and you only want to break it if it's a small scale to start with. Hence, a demonstrator rig at 2 million gives you a size of the problem. We're only going to 1 meter cubed. If anybody wants to know what that is, that's the black bins we used to have outside our houses. Which is
Okay.
Scale. Yeah?
Probably ought to move on. We probably can talk about this another time. So,
Indeed.
Lucan Gu has said, do you have a general sorting before hydrocracking to separate different molecular weights of plastic waste?
Oh, good question. If you think about the answer to the question, is the whole ethos of the project was to take polyolefins, don't care what they are, don't care what molecular weight they are, and turn them into a useful product. Obviously, if you think about using polyolefin resins as manufacturers would make, the molecular weight is very, very important. And you're just adding to the complexity of the problem. If you go to the man at Veolia or the lady at Veolia and ask what their problem is, they've just got a dump truck that's dumped everything in it. So to sort them out would take a long time and is very cost effective. It's one of the stumblers. Therefore, I think the way forward is to look at bespoke units, maybe 5 to 15,000 tons per annum sort of units on specific sites, which are already designated. Calagas is one of those that would know all about being a chemical site. So we haven't sorted on molecular weight because we didn't want to initially, because we wanted to prove it would work with anything.
Okay, great. Patricia says, do you have a good knowledge base that the hierarchy you showed is consistent with the environmental benefits, particularly carbon, that would be delivered by taking those approaches, i.e. do the top options save most carbon and the bottom ones save the least, or does it vary?
Oh, that's a good question, Patricia. I don't know that I could answer that straight off the top of my head without getting it wrong.
That is a good question for us to have a look at, though, I think.
It is. I absolutely agree, and that's why I think it was. I was going to say that. Thank you, Chris. So yeah, that would be good. That is something to look at, and that is a way of because we the hospital waste that we talked. I just showed briefly that waste up there before. That's part of this RE3 project, and that has brought, oh, well, I wouldn't say open my eyes, but has brought in a systems approach to look at how flows of things flow through the system and how they might be treated. So I think it's a good thing to look at, yes.
Yeah, okay.
Somebody write that question down.
Yeah. So Jonathan Wagner says, have you done any assessment of the impact of biopolymers and biodegradable polymers on the cracking of mixed plastics?
Okay, so I don't know the affiliation, so I don't want to offend. Biopolymers are about 1% of the total product stream. And so the answer to that is no, we haven't. Although using certain oxygenated polymers and adding them into the mix has some advantages with regards to selectivity and processing ability. So for example, adding PET into a polyolefin mix has some benefits to product selectivity, but it's no more than lab scale. I'm not sure I answered the first part of that, Chris. Is it possible to repeat?
Well, I think it's mostly, you know, if we have a mixed polymer stream where we're looking at polyolefins with, I don't know, polylactic acid, for example.
Yeah.
What's the impact? There will be quite a significant impact because most of the biopolymers are oxygenated, I guess. So there will be an impact in terms of production of water, for example, in the hydrocracker and so on. So
that's true, and that's what I was effectively. That's what I was alluding to.
It would be interesting to do, though. I think so.
It does, and steam and CO2 are actually very useful on controlling selectivity of cracking reactions, as people know in the production of LAS, linear alkyl sulfonate. Sorry. So if you're cracking something over alumina, if you add CO2 or water to it, you can reduce the number of side reactions significantly. Something like 150 side reactions down to 25, something like that.
Yeah. We're just going to do one more question because otherwise we're going to run out of time.
Sorry, everybody.
Devaduta, how much yield difference is there observed between the thermal and catalytic cracking of waste plastics, have you got any hard, solid products in the catalytic pyrolysis? It's not really pyrolysis, I guess, that we're doing.
No, it's not. No. So yeah, so thermal cracking just gives you everything. So you will thermally crack something, and you will get a range of very broad range. So the advantage, apart from the fact it's very hot and using a lot of energy. So thermal cracking, 700 degrees sort of temperatures, 500, 700 degrees processes, whereas the hydrocracking process is operating at a much lower temperature. And the use of the catalyst, even if they be NEMO or COMO type catalysts in hydrotreating, your range of products is going to be narrower to gas and liquid. So LPG, liquid, like naphtha and diesel sort of aviation fuel level. So the issue is not about the conversion. Our conversion is everything can be converted. If you're trying to scale something up, you've got to reduce that conversion so you can actually look at the kinetic of it and try and design the reactor. So thermal will go. It's just it's very hot, very bad for the environment because you're using a lot of energy. I'm not saying that we're not using a lot of hydrogen. So before somebody jumps on that, but you can. It's how you source your hydrogen. And that's where there's another linkage, I think, to the SuperGen bio hub.
Okay, that's great. And as Patricia said, I think this is a good way of trying to link the two hubs together, as I think as well. So
yeah, I did see Matt Davidson's comment pop up there as well. So I agree entirely. Yeah.
Okay, brilliant. I think we should carry on, but in the meantime, thank you, Arthur.
Thank you. Thank you.
Right, and last but not least is a colleague of mine from Belfast. Hello, Haresh. He's going to talk about catalyst design and process optimization for sustainable production of biofuels. Take it away.
Can we see the slides? Hello?
Yeah, we can see the slides, it's fine.
Okay, so I'll carry on now. Right, as Chris said that we'll be looking at the opportunities to link between the Catalysis hub and the SuperGen Bioenergy hub. And this is what I'm going to show. Some of the work which we have been doing in Belfast is exactly providing the bridge between the two hubs. We'll be talking about catalyst design and process optimization to produce the biofuels. As we can see that UK is the first major economy that has already committed to the net zero target, which is the most ambitious target to achieve in the world. To do that, the conversion of the biomass to biofuels will play an important role in this area. However, to achieve that sustainably, to do that because plants grow and they absorb CO2, they consume CO2, and then while we burn the fuels, the CO2 is liberated again. To do it sustainably and to be able to do that more efficiently, what we need is we need the conversion of the biomass to biofuels with very high efficiencies. We need the mild reaction conditions, we need high conversions, and we need high selectivities. So we can do that by using a variety of different feedstocks from biomass, which will be based on lignocellulosic biomass, which can be pre-treated in different ways to utilize it. Also, we can see that the International Renewable Energy Agency's roadmap tells us that by 2030, we should be doubling the share of the renewables, whatever we are contributing in terms of the biofuels, in terms of the diesel and the different forms of renewable energy. However, the previous targets which were set for 2020 were up to 10% of the biofuels should be blended into transportation fuels. But currently, based on the recent statistics from the Transportation Department in 2019, was that UK is meeting only 3% out of the set 10% of the target. And now we are looking at doubling our contribution. So how we can achieve that? This is the big chart of different types of components which are present in the biomass. We can look at cellulose, we can look at hemicellulose. There is also a variety of feedstocks, which we call it as oils, vegetable oils, triglycerides, which can be converted into diesel and biodiesel again. And we have been working on a number of these target molecules in Queen's University in Belfast, looking at things like hydroxymethyl furfural or furfural-based furanic feedstocks, or working on triglycerides and glycerol type of feedstocks to convert them into fuels and fuel additives. So these are some of the exemplar studies in which I'll be talking about. So it's kind of an outline of the talk today. So we will be looking at conversion of HMF to BHMF, which is the fuel additives. We'll be looking at conversion of vegetable oils and fatty acids directly to hydrocarbons, which is again called as green diesel. And we'll also be looking at how can we directly convert biomass, which is cellulose and grass silage, to make fuel additives, which are alkyl levulinates. And in the end, to close, I'll be talking about glycerol valorization into glycerol acetons, which are again fuel additives. So as we can see that HMF is one of the important furanic ring, which is derived from sugars, from carbohydrates, and that's also known as platform chemical or a sleeping giant, because it can be converted into a huge range of chemicals and fuels, which have got a variety of applications. So that's that can be seen here on this slide. I will go to the particular reaction that we are studying in Queen's in Belfast. That's about selective reduction of the HMF molecule here to convert it into BHMF. BHMF is useful as a fuel additive, and reduction of the BHMF has been studied extensively in literature. There's a number of studies that have been performed in literature before with very good and very high yields of BHM. However, if we can look carefully, the reaction conditions are far from being able to scale up very easily and economically. What we need is the process which is energy efficient and the process which we can scale up easily. So the catalyst should be commercially available and economically viable for the scale up. The number of catalysts which currently are there out in the literature are specific and require synthesis, which makes them difficult at the large scale. The catalyst which we have been working out with this conversion is based on manganese oxides, and manganese oxides are known as reducible oxides. They perform very well in hydrogenations and oxidation reactions. We have also carried, these are nano rods which look here like this in the TEM image here, and which are sprinkled with platinum nanoparticles at the top like a fairy dust on top of it. So the catalysts were evaluated for the reactions. We got very good conversions and very high selectivity and very high yields to BHMF. Close to 95, 96, and 100% conversions were achieved, and 80, 90% of the yield to the product was achieved. We studied the reaction conditions were optimized, process was optimized to make sure that the reaction was free from the mass transfer limitations. So all the catalyst particle sizes were very small, and we got a very linear response to the catalyst loadings. And we studied the effect of temperature and energy activation here to make sure. This is a quick slide here to show that we did look into the mechanism of the reduction of the aldehydes and ketones before using density functional theory calculations as well. And the reducible support manganese oxide itself can participate in the reaction as a catalyst. So it acts through the water-assisted process where water is dissociated on the surface to provide the hydrogen for the reaction. In this case, this is a similar reduction of aldehyde again to alcohol, in which we are converting HMF to BHMF. So platinum mono MS2 catalysts performed really well. Catalysts were recyclable and reusable. Very high conversions and selectivities were obtained, and we could look at this catalyst for techno-economic analysis for scale up of this process. The other part of the study, other example which I'm going to show, is direct conversion of the oils into hydrocarbons. Again, because converting oil into hydrocarbon is quite an important technology here, because we can look at non-food-based inedible oils like canola oil or soybean oils, and we can also look at the used cooking oils which can be converted to green diesel or hydrocarbons as a feedstock. Normally, the reduction of the oils is very difficult. It requires very activated catalyst. So we designed this catalyst. Again, this was inspired by theory calculations by Page and Hu's group. We did a bimetallic catalyst, platinum, rhenium on titania, and using this catalyst, we were able to achieve very high conversions and selectivities. We started this study with triacetin as the model substrate, and we could selectively convert with almost 100% conversion of the triacetin with 99% selectivity to alkanes, which are very useful as green diesel. We looked at a variety of different oils like palm oil, sunflower oil, olive oil, corn oil, coconut oil, and all of these using bimetallic catalysts, we were able to convert with very high selectivity to alkanes to produce biofuels that can be used here. We did extend this study to stearic acid as a model substrate to study the catalyst reusability. The reusability dropped down slightly there, but which could be mitigated very easily by using the continuous flow reactor. So this is the continuous flow reactor that was designed in Queens, which is, it can produce very easily up to kilos of the fuel per day. And this is the stability of time on stream of the catalyst, which indicates that we did not lose any interface between the bimetallic catalyst, and very high conversion and selectivities over a steady period of time was achieved using this. So bimetallic catalyst will be used for conversion of oils directly. In the next study, we used conversion of biomass derived grass and cellulose, and we also started with furfural and furfural alcohol to convert it into butyl levulinate. So butyl levulinates are alkyl levulinates, which are very good fuel additives, high energy density, which is very similar to the regular gasoline. It is much better than ethanol, and it's good for reducing the greenhouse gas emissions as well. So again, in literature, the catalysts, which are very sophisticated catalysts, which are difficult to scale up, and that is why we decided to go back to economically viable catalysts, which are easy to scale up and easy to procure it commercially. So we looked at heteropolyacids. Heteropolyacids are strong acids. Bronzed acidity is very high in it, and it has practically no corrosivity. We can handle it by hand as well. And has some disadvantages because it's highly soluble in solvent, but can be mitigated by immobilizing the heteropolyacids on the surface of different supports. In this particular study, we chose ketane as the support. Monophenolate ketane was chosen as the support. Using this, we did make the catalyst by mobilizing it on the support, and the protons of the heteropolyacid were displaced with tin to enhance the acidity on the surface of the catalyst. This screen shows the reaction scheme where we can convert furfural alcohol to butyl levulinate. It's a two-step process which passes through the formation of the ketone as an intermediate here. However, it can be converted into a one-pot process where it all happens in one reactor. So this is the reaction scheme profile as a function of time, which shows very high selectivity to N-butyl levulinate, and very high conversions of furfural alcohol were achieved in this process. Again, the catalyst and the process conditions were optimized to achieve a scalable process in a temperature ratio, and the reusability of the catalyst is very high. Also, the hot filtration test for this to check the true heterogeneous nature, so that we are sure about that there is no leaching from the surface of the catalyst here. And the reaction was performed using cellulose as a substrate as well, because furfural alcohol is normally produced from cellulose to sugars, and sugars to furfural-aldehyde, and which is then subsequently converted into furfural alcohol. So what we did, we directly started with cellulose. This was done as part of the CaviCat project, in which we combined it with ultrasound for the pretreatment of the biomass to break down the biomass. And in this reaction, again, starting with cellulose, in combination of the catalysis and the cavitation phenomena originating from the ultrasound, we were able to achieve, again, very high conversions of cellulose and very high yield of butyl levulinate in this case. In the last study, I would like to talk about conversion of the glycerol to fuel acid. Glycerol is the byproduct from the formation of the FAME, which is fatty acid methyl esters, which is known as biodiesel from oils. And it's almost 10% of the biodiesel that is produced by weight. So what we did here is we did the esterification of the glycerol with acetic acid to convert it into diacetins and triacetins. And again, the process requires efficient catalysts which are scalable and economically viable and recyclable. So this particular study, we chose Bronsted acid ionic liquids. Range of different ionic liquids were studied, and NMP HSO4, which is pyrrolidone-based ionic liquid, gave us the best results in terms of the conversion and very high selectivity to di and tri. The process was optimized using design of experiments approach, in which we optimized various different reaction parameters in terms of catalyst loading, temperature, and mole ratios. And it was observed that there was a synergy between the mole ratio and the temperature of the reaction here. We did look at the reusability of the catalyst, which was again excellent, very high conversions for reusability of the ionic liquid were achieved. Ionic liquid was in a separate phase, so it was liquid-liquid mixture. It was very easy to separate and very easy to recycle. And the catalyst is economically viable for scale-up. All this work was done in collaboration with a number of colleagues, so it's a big team effort. I would like to acknowledge their efforts in terms of helping for doing experiments, for providing a lot of valuable insights into it. I would acknowledge Chris Hardaker, Professor Pajan Hu, and Laura Catherine did the work based on the acids, and Manish did work based on the butyl levolinates. John did work on glycerol. And this is the team here in Queens. We have a small group here. And last but not least, I would also like to thank all the funding agencies for supporting this work, which has been through InvestNI's Center for Advanced Sustainable Energy and Catalysis Hub and EPSRC for different projects. And thank you to everyone for listening to this talk, and to organizers for giving me opportunity to present some of my research here. And if there are any questions, I'd be happy to take them.
Thanks very much, Haresh. Are there any questions for Haresh? I can ask one. And in terms of the overall yield that you were able to get of your hydrocarbons from your fatty acid methyl esters, what sort of yields were you able to achieve in the end with your continuous flow reactor?
Somewhere between 90 and 95, we were able to achieve 90 to 93% yield to hydrocarbons.
Is that with a single pass, or was that with multiple passes?
Oh, I get it. Sorry, 92 to 93% yield was achieved in the batch reactor. In the flow reactor, it was close to 52% conversion was achieved under optimum conditions.
With a single pass?
Yeah. With a single pass, yes.
Okay, okay, thank you.
Yeah. But the advantage here was that we were able to use very high concentrations. So we were able to use 50% of the substrates along with the solvent ratio.
Okay, okay. Thanks. Any other questions for Haresh?
Haresh, it's Mike Bowker. Of course, in hydrogenations, you're often using precious metals, as we have in our work too.
Yes.
Do you see any prospect of getting away from these non-rare earth abundant elements to more abundant elements in catalysts for these kinds of processes?
Yeah, there is, we are looking at using the metals such as copper and nickel, and currently we have got excellent results from the project students which are looking at reduction of levolinic acid. I couldn't show the results today here, but we have looked at copper supported on manganese oxide as a very good catalyst, because the idea is I would like to go towards a commercially available catalyst such as hopcolite, which we can buy it, which is again a combination of the copper and manganese oxide materials. So that's what we are doing at the moment. And the manganese oxide itself is a reducible oxide catalyst, which can do the reductions on its own without any additional metal onto it. but the reaction rates are quite slow. So to enhance the rates, we do need a metal which can then give us a spillover effect with hydrogen by dissociating it as well. So what we did is we started looking at copper on manganese oxide, and it is giving us very good results for reduction of levulinic acid to GVL.
Excellent, excellent. Well, good luck with developing those kind of things further and getting rid of the precious metals as much as you can. I don't think we'll ever be able to get rid of them entirely.
No, but we can minimize the use of the precious metals, and that's what we say, that we can use them more efficiently. So if you can see that instead of conventionally using 5% or 10% of the metal, now we are down to using 1% or 2% or 4% of the metal supported with reducible oxides.
Yeah, yeah, very good.
Okay, there's a question from Jude. Are you able to say what the fractional alkane yields are?
Yes, we can, depending on the composition of the oil which we are using, it varies between C10 to C16 or C17. And by looking at the calibrations through GC, we can definitely say the fractional alkane yields.
Okay, that's great, thank you. Okay, there are no further questions, so I'd like to thank Haresh again. And thank you to all the speakers this morning. And I forgot to say a special thanks to Josie, Corinne, Nathan Skillen and Helen Daly, who actually organised this meeting today. So thank you very much indeed to you for all of your help and efforts and everything else like that in putting this together. It's a wonderful programme. We're on a lunch break now, and then following the lunch break at 12:50. We start with Jason Hallett. So we're on the same Zoom, I think, aren't we, Josie? Is that right?
Yes. Yes.
Okay. So
I'll leave it open in case anyone wants to have a chat over the lunch break or anything, but I'll pause the recording.
Okay. Brilliant. Thank you very much.
So dream of telling you of anyway. I might talk about you.
Right, so welcome back everybody. Thanks for sticking around to listen to the second half of today's session. I think we can already see that there's a lot of work that bioenergy and the Catalysis group can do together, and I think that that's only going to increase as time goes by. So these sessions are great because we can get so many people listening to our work and hearing about what we're doing, but they're not so good because we can't actually talk to each other. So I'd just like to get to know who's out there. So where are you all? So I'm Katie, I'm in Birmingham, and I'm in my spare bedroom. So if you could just go on the chat and tell me where you are, I'd like to know who's the furthest person away from us. So I know there's people in Manchester. Jason's in London. Are you in London, Jason?
I am indeed in London.
So, oh, Patricia's here in Cheltenham, Cheshire, Belfast. Have we got anyone further afield? York, Reading, Belfast, Brazil! We have a winner! Saudi Arabia, wow, Indonesia, Dan in the dining room with his dog, I bet. Hi, Garay. I haven't seen you for a long time. Singapore, wow, okay, so this is fantastic. We've got people from an Alf the cat, of course, Nathan. Excellent. I like to know that there's lots of pets watching, or I also enjoy it when pets join in on the presentations. Okay, so we've got people from all over the world, so that's one of the brilliant things about doing these things online. So welcome everybody, and now I'm going to hand over to the one and only Jason. As before, if you have questions, put them into the group chat. If we've got time, we'll have them at the end of the presentation. If not, we'll save them till the end. Okay, so over to you, Jason.
Okay, thank you, Katie. For those of you that don't know me, I'm Jason Hallett from Imperial College London. And I am one of the SuperGen investigators working on, in my case, pretreatment of biomass. And I'm going to focus most of my presentation on the pretreatment activities at a very high level. So I've stuffed all the results into 2 slides, as you do. And because this is a Catalysis hub, I flipped a coin as to whether to talk at the end for 5 minutes on chemocatalysis or biocatalysis, and we're going to go with biocatalysis. So we'll do a very brief examination of some of our biocatalysis work too, just to keep the theme alive. There we go. My slides are a little slow. So I use ionic liquids for pretreatment, and as with anything, when you're doing something different from the norm, or for that matter, whenever you're using ionic liquids, I feel like you need to have a fairly good motivation, because they can be quite tricky to work with in some instances. If you're looking at pretreatment as a first technology or first step in the transformation of lignocellulosic biomass into sugars, and presumably for biofuels, the most important aspect of a pretreatment is to get the highest enzyme activity in the second step, and therefore the highest fermentation yield at the lowest cost possible. And there are a lot of pretreatment technologies out there, and a lot of them have suffered, rather than in a technical sense, but in an economic sense. The reason that I became fascinated with the use of ionic liquids for pretreatment is that they work better than everything else. And the Joint Bioenergy Institute in the US has done a lot of work on ionic liquid pretreatment, and as part of the big DOE study on pretreating various biomasses, they've done 28 different biomass types with, I think, 11 different pretreatment technologies, and the ionic liquid has won 28 out of 28 times. So it works really well. The downside to them, while they're very efficient, is that they're very expensive. And so my research early on, 5 or 6 years ago, was focused on trying to flip that around. So since ionic liquids are basically just salts in the end, and we all know that salts don't have to be expensive, can we find an organic salt that will take biomass apart without costing a fortune? So obviously the answer is yes, or we wouldn't be here. But how did we get to that? The biggest thing that I had to throw out the window in order to do that was the biggest advantage that ionic liquids have over other solvents, and that is that they dissolve cellulose. Back in 2002, Robin Rogers discovered the dissolution of cellulose into ionic liquids, and later on, people have increased the solubility of cellulose in ionic liquids as high as 28% in some cases. So they're outstanding solvents for cellulose dissolution and decrystallization, so they make amorphous cellulose. And this is great if what you want to do is process cellulose. Unfortunately, in order to get the cellulose into the ionic liquid, you have to dry to less than 0.25 weight% water. And if you're intending to process biomass, the odds of you having wood that has less than 0.25 weight% water is pretty much zero. So you have to dry the wood forever, and you have to dry the ionic liquid forever to make this work. So my thinking was, instead of trying to dissolve cellulose, could we basically run an organosolve extraction? And so do an organosolve lignin extraction using an ionic liquid. And in that case, what you're looking for is a solvent that is an excellent solvent, an ionic liquid that's an excellent solvent for lignin rather than cellulose. The big advantage there is that it gives us a much larger space to play in. And ionic liquids people like me are very fond of bragging about how many billions and trillions of ionic liquids are possible. But there's only 3 that really dissolve cellulose. So I want the rest of those billions and trillions of possibilities. And so what our ionosolve lignin extraction process does is it takes biomass, it dissolves lignin and hemicellulose, much like an organosolve pretreatment, and you get a high purity cellulose pulp that you can filter off. And you can see some pictures there of our lovely products. That is an unbleached pulp, actually. And so the question is, do these salts have to be expensive? And the origin of the idea that ionic liquids are very expensive, as far as I can tell, dates back to the days where it was used as a fuel cell, fuel cell electrolyte. So this salt at the top, butyl methyl imidazolium bistrifluoromethylsulfonylamide, you can say that very fast, is an extraordinarily expensive solvent that's been proposed for use in fuel cells. But it's not the one that you would want to use to take biomass apart. And so I was inspired by the idea that if we combine mineral acids with alkyl amines, that we can make much cheaper salts. And this is actually an even more aggressive way of doing the phase transfer catalyst type synthetic methods. And so instead of these sort of 2,000 pound a kilogram type of salts, or even the deputetic solvents, which run around 50 pounds a kilogram, we have made ionic liquids, and we've published a lot of techno-economic studies on this, that are around 50p a kilo or 500 pounds a ton. And for reference, it's about half the price of acetone. And so they are moderately low cost organic solvents. Actually, it's almost identical to the cost of ethanol at the moment if it's used as a solvent. So that part was fine. And so here's one of the slides where we stuffed all the results into one slide. So how well did it work? You can see some of our cellulose pictures in the upper left-hand corner, sort of the visual images, which I always like. So there's some nice pretty white cellulose that we isolated from waste wood. And you can see in the upper right, the SEM images where we took that cellulose and filtered off the suspended fraction of what turned out to be cellulose nanofibers out of the ionic liquid. We get about a 5% conversion to cellulose nanofibers automatically during pretreatment. A lot of the aspects of the ionic liquid were good, some of them not so good. One of the nice things about it is the more you recycle the solvent up to about after about 4 or 5 cycles, the sugar yield increases and then levels off. So the lignin chemistry actually evolves quite a bit, and the dirty solvent is better for extraction than the clean solvent. It's very stable, so these ionic liquids are quite thermally stable. At 330 degrees, they boil. But up until that point, they sort of stay put. You don't get any breakdown products. You don't get any inorganic salt dissolutions. They're quite poor solvents for inorganic salts, which is useful. You don't want to have a buildup of salts in the system. We recover the lignin by adding a small amount of water to the ionic liquid to precipitate the lignin. We published a study with SuperGen earlier this year. You can see the graph here that showed that if instead of just adding a bunch of water and precipitating the lignin, if you add it slowly, you can fractionate the lignin and you can tune, in this case, the thermal properties of the lignin, but actually it's the molecular weight and the subunit linkages that are most affected during fractionation. So you can do lignin fractionation as part of your pretreatment technology rather than have to do a separate step to do it. We've been working a lot recently on intensification. You can see the graph on the far right, where we increase the temperature to reduce the residence time from hours to minutes. Haven't gotten quite down to seconds yet, but we're at about 2 to 3 minutes at 180 Celsius. So you can have quite quick pretreatments that are very efficient. And you can see in the lower right the compositional analysis of the ionosolved pulps. And the pre-bleached pulps are quite similar in composition to dissolving pulp. And actually, in some cases, when we do the recycled experiments, we can get ionosolved pulps with very similar compositions to cotton. So quite nice, quite pure products that we get out. The economics are very good, and I don't want to dwell on economics because honestly, it's a large portion of what I do in this space, but actually the end message is much more exciting than the methodology. The bottom line is if you take, in this case, it was an energy crop we did for this study. We took miscanthus grass and we calculated if you run it through this process and you run it through the recycling of the solvents and you run through all the downstream separation units, you can produce glucose from miscanthus for about 14 pence per kilo, which is roughly 2/3 the trading price of food sugar. At the time, it's actually less than half the trading price of food sugar right now. So that's quite useful. It's a very intensified process. You can run it very quickly. You can make a lot of interesting products, either cellulose or sugars. And if you wanted to use that for biofuels, it's certainly viable, shown that the fermentation is very ready. From there, we moved on to looking at more interesting feedstocks, in particular waste wood, particularly wood that's been preserved with either heavy metals or we also do creosote treated wood. And looked at how the metal extraction is affected. Since the ionic liquids are salts, they're quite good complexing agents for a lot of metals. And my graph there got cut off a little bit, but you can see that the metal extraction generally is quite good. The copper extraction is basically quantitative, and we can take things like chromium and arsenic out in around 80 to 85% quantities. We took that further and tried to do our pretreatment on post-consumer waste wood. And so the two graphs at the right show how much sugar we can get out of post-consumer waste wood. So this is basically, fencing and that sort of thing that's been torn down. And in the bottom right, how much metals we can get out. And there's all sorts of metals that we've looked at, everything from lead to cadmium to nickel. And the metal extraction, again, very good. It's generally higher than 90%. And the sugar yields at low biomass loading, so low for me is about 10 weight percent biomass, are quantitative, and they do decrease at higher loadings. And so there's an economic trade-off to be taken with how much that you do. This was quite an interesting study because it took advantage of one thing that ionic liquids do that other solvents don't. So it's very difficult to find an organic solvent that will complex metals and fractionate biomass at the same time. And even in water, this would be impossible. So we've done, we spun out a company which has been renamed now, so it's now called Lixia, to look at the scale up. And so we've demonstrated this up to the 200-liter scale. It's about 50 kilos of biomass. And at the moment, well, not the moment, but in about a month, we're going to start construction on a pilot plant in Sweden to bring this process to commercial scale. So to conclude the bit on pretreatment, I always say if there's one thing to remember from what we've done with our work in using ionic liquids for biomass fractionation, it's that the salts don't actually have to be expensive. You just have to think about what you really want it to do. In my case, I decided that if it was made half out of sulfuric acid, that it would definitely fractionate lignin and also definitely be cheap. And so this delignification process, as opposed to the cellulose dissolution process, it's quite exciting because we're only doing lignin extraction. We can use much higher solids loadings, and so we don't need as much solvent in order to get the same performance. Now, the last thing I wanted to mention, and I did manage to leave myself a few minutes for it, what I haven't talked about is the downstream processing. So after the pretreatment, what are we going to do? And one of the things that we wanted to look at was how the ionic liquid might impact the enzymatic activity if you wanted to do saccharification with cellulases. And eventually we were thinking, well, how could we sort of combine these two steps into one? And this was an idea that we kicked around for a while. So if we flip this around, since you do have ionic liquids that could dissolve cellulose, if you wanted to dissolve cellulose, could you stabilize an enzyme enough that it would be able to act on cellulose in an ionic liquid with no water present? So this is completely non-aqueous biocatalysis. You can't immobilize cellulases because you end up with solid-solid reactions. So you have to use soluble enzymes. So we started looking at different techniques and developing different techniques for stabilizing enzymes using the ionic liquid in place of water. And what has worked the best for us has been the area of liquid proteins, which we've published quite a few papers on now, maybe a half dozen. I put 3 of them in the lower right if you want to learn more about liquid proteins. And what happens is you basically run the EDC reaction on the surface of the protein to create either cationized, you can also do it with an anionized charge layer, and then you use an ionic surfactant to stabilize the surface. And the nice thing here is when you put this into an ionic liquid, of course, you get a separate electrostatic stabilization of the fold. And so now instead of hydrogen bonds locking the protein fold in place, you have coulombic interactions locking it into place. This completely prevents the proteins from aggregating, which is quite important. And so we were able to show that you can get up to 50 weight % protein into an ionic liquid without it aggregating. And I'll have some pictures of it there. And we've done a lot of protein NMR studies to figure out where exactly the ionic liquid interacts with the proteins in order to keep it so stable. The thermal stability of these is quite impressive. For some of the enzymes that we've used, it's been as high as 200 degrees. The ones I'm going to talk about today were pretty good up to about 140 Celsius. And again, this is a soluble enzyme dissolved in ionic liquid that is less than 0.2% water. And so it's a very, very anhydrous medium. And the idea here was that we could switch from this sort of very complicated cellulase cocktail that's traditional, where you have the endo and exocellulases to break up cellulose, and then a beta-glucosidase that comes in and chews up the cellobiose dimers to release glucose. Our thinking originally was that in the ionic liquid, you could just dissolve the cellulose. And this would be quite good because now all of the enzymes are acting on a soluble substrate rather than an insoluble substrate. So rather than the anchor coming in and the solid-solid interaction in water, instead what you have is a quite powerful solution phase technique to do the same thing. And you can read more about it. We published this 2 years ago. Fortunately, actually it worked quite well with any of the enzymes that we used. And so the one that we took forward in the study, because it was quite a quirky little thing, was an alpha glucosidase. And so rather than stabilize all of the enzymes in the cocktail, we found that actually you only need the glucosidase. Because once the cellulose is dissolved in solution, the glucosidase enzyme can now bind to the end glucose units on the cellulose chains, which have, of course, been defibrillated inside the ionic liquid, and can chew off glucose monomers directly. And so that reduces the enzyme cocktail from 3 enzymes to 1. And it also enables you to use basically any glucosidase you want. We even showed it with an alpha glucosidase, which is not a normal substrate for any of these. And you can increase the temperature to increase the activity. And so we were able to run these at about 110 degrees C. And we saw absolutely incredible activities. They're about 30-fold increased over on cellobios, which is the normal substrate. They are infinitely more active on cellulose because glucosidases have no enzyme activity on cellulose. So I want to conclude there. What our final vision for that sort of catalytic process is actually even more simple than what I said before. You dissolve the cellulose in the ionic liquid, you take 1 enzyme and you release glucose directly. And so that's quite a quick way to make glucose from cellulose. And so this was work that we've done in Imperial College. The enzyme stuff was funded by the EPSRC, the scale-up stuff by the Royal Society, and of course the pretreatment by SuperGen. So thank you. I'm happy to take questions.
Thank you very much, Jason. Great presentation. It's nice to know that you're employing wizards. You're saying that Alex is a sorcerer?
I am. I mean, I absolutely incredible. I mean, I joke about this sometimes, but you're not supposed to be able to do what we did. So you're not supposed to be able to depolymerize cellulose with 1 enzyme, and certainly not that enzyme, because that enzyme doesn't bind cellulose.
Yeah, that's fantastic work. So I've got a couple of questions. I think we've got time for one before the next presentation. So will some of the lignin, or chemistry words, moieties be aminated, particularly on benzylic parts, which is easily activated by acids?
So this is a good question. So I wondered about this. So if you take a weakly complexed acid-base ionic liquid, so a protic ionic liquid. So if you take an acetic acid amine ionic liquid, the answer is yes. We use sulfuric acid amine ionic liquids, and so there is no free amine. I mean, you can calculate it out, but the pKa difference between those two is 14. So it's not, there's no free amine in that solution. And so we don't see any amination because there just simply isn't any free amine in solution to happen. We have scoured our lignin and our cellulose in each experiments with the most intensive CHNS we can possibly find to see if we're getting any nitrogen complexation. We have never seen any to date with hydrogen sulfate ionic liquids, but you do see them with weaker acid.
Thanks, Jason. That made no sense to me because I'm an engineer, but I hope that made sense to the person that asked the question.
It was a perfect answer, of course.
There's some other questions. We've got a question from Sanjay and another question's just appeared. So if you could answer those while we...
Yeah. So the particle size question. So I'm sorry, Sanjay, that was actually in...
Not now, we're moving on to the next
okay. Yep.
So I'm going to move on now to the next one, so we stay on time. So thank you, Jason. And now we're moving on to Sanjay. Sanjay, if I can hand over to you.
Yeah, thank you very much.
Yep, ready to go whenever you're ready.
Yeah, okay, I'll just share my screen now. So can you see my screen now? Yeah?
Yeah, we can see that.
All right, okay. So what I'm going to talk about today is, first of all, I'm Sanjay Nagaraj from Queen's University Belfast, for those who don't know me. So what I'm going to talk about today is cavitation pretreatment for enhanced biogas generation. And it makes it easy for me to just skip the slide because people have been talking about bioenergy and renewable energy since morning. So what I want to just focus on in this particular slide is nothing but saying that we've got 2 kinds of biofuels, either liquid biofuels or gaseous biofuels. And we've got a first generation biofuel production mode and then second generation mode as well. So first generation, obviously we've got problems with it. We can't use the land that we use for food crop production, and then we can't use, of course, the food materials as such as biomass for bioenergy production. But second generation, on the other hand, we could use waste biomass resources as feedstock for biogas or biofuel generation in that case. With liquid fuels coming all the way from first generation towards second generation, gaseous biofuels, specifically biogas, is always concentrated on using waste feedstock as a source for bioenergy production. This can include from municipal solid wastes, sewage sludge in the wastewater treatment industry is quite common. And then we've got food waste, agricultural waste, or even forest residues. So with that, I will move on straight into talking about what AD is for our non-bio friends there. So AD is nothing but anaerobic digestion, which is a technology that is exploited for the production of biogas. And biogas is a mixture of methane and carbon dioxide predominantly, and we've got other trace gaseous elements as well there. Gaseous molecules there like hydrogen, nitrogen, meager amounts of oxygen, hydrogen sulfide if we've got enough sulfur content in the biomass, and then ammonia as well in some other cases. So what happens in anaerobic digestion is the feedstock that we pump into the digester is acted upon by complex synergetic microorganisms. The microbiology itself is quite complex, but the synergy works quite well. And that is why AD is one of the most robust processes. Of course, there's argument that when a digester fails, it's difficult to put it back on track, but it can actually get it back on track with time. With manual interference, of course, it can be done much more quickly, but AD is quite cheap. It is robust technology, so people have been focusing on it for a while now. And AD has been an established technology for quite a while for not only just reduction of COD of wastes in terms of wastewater treatment industry, but also for the production of bioenergy as well. AD by itself can act as a biorefinery because we don't only just produce biogas or a biofuel in that sense, but we also end up producing digestate, which could be used as fertilizer. Of course, we've got regulation that control the digestate spread on the fields. But other than that, we could still use digestate to maximize the potential of the feedstock itself, which I'll be talking about a little bit on later in my slides. But predominantly, this talk is going to be about how the pretreatment technology can enhance the biogas production. So biogas production is predominantly affected by the feedstock type. So, you know, AD is quite robust technology. It is quite good. It's established. It's worldwide. But there are two major problems with AD. One is the retention time. It is quite slow because methanogens are quite slow growers. They can take from days to a few weeks to multiply. So because of the metabolism and growth being too slow, the retention times have to be in range of weeks at times to get enough conversion. And then the conversion itself of the feedstock towards biofuel production is quite low. So with complex lignocellulosic feedstock for that sense, there's about 50-60% conversion of the feedstock to biogas. In terms of sewage sludge, when used as feedstock or food waste, for that case, can go up to 80-85%, which is not an issue. But again, because we are talking about agricultural residues are predominant feedstocks in the modern days, we would want to ensure that the conversion actually is maximized, which means that will impact the digestate quality and the fertilizer that we produce at the end as well. And in another case, if we don't want to use the digestate as fertilizer, we could probably extract the organics out of the digestate and then further process it towards other processes, which I will again talk about later. So first, talking about the feedstock itself, the feedstock is quite complex in terms of agricultural residues. We've got cellulose, hemicellulose, and lignin as the predominant components. But in other cases, for example, food waste, because the feedstock is quite easy to act upon, the rate limiting step actually differs in this case. So in anaerobic digestion, there are four predominant steps. The first step being hydrolysis, which is nothing but breakdown of the complex polymers to monomers, soluble monomers. And the second step being utilizing the monomers towards acid production. And third step being acetic acid production from these volatile fatty acids. And finally, methanogenesis. Methanogenesis can be either acetoclastic, meaning the acids are utilized towards biogas production, or hydrogenotropic, meaning the hydrogen and carbon dioxide produced in the digester is used towards biogas generation. So when the feedstock is quite simple, it can be hydrolyzed quite quickly, or whether if it's available as monomers itself in the case of food wastes, what happens is we tend to acidify a digester quite quickly. And what happens there with an acidification of digester is there's an imbalance in microbiology, and we will not be getting enough biogas. We'll probably end up with a lot of acids and hydrogen and carbon dioxide. So we don't want that to happen. So for that case, the rate limiting step will be hydrolysis. So we need to consider retention times being too low and balance out on the organic loading rates for that sense. In terms of agricultural residues where it's lignocellulosic biomass that's being fed to the digester, we've got the rate limiting step as the methylogenesis step because of the multiplication and metabolism of the methanogen itself. For typical industrial digesters, we talk about feed loading of about 10 to 15% solid loading. We can't go above 25% because it's a pumping limitation, and current pumps can pump slurries up to 25%, but not beyond. And 25% being the maximum is never a good idea to do it with digesters because we don't want that to limit the process, which can actually just increase the retention times beyond 4 weeks. So we want to keep it to a moderate amount. So 10 to 15% is usually a norm in the industry. And then temperature affects the microbiological process, not just this, but any microbiological process. So mesophilic is the most common ones, which means digester is operated at around 30-35 degrees Celsius. Thermophilic is anything above 45 until 60 degrees. And cyclophilic is cold temperatures, which is below 10 degrees, which is a new area of research, but mesophilic and thermophilic are quite established, and there's more focus in this area to maximize the outputs of biogas. I don't want to go into other details, but what I would want to say though is every other detail that I've mentioned in this slide is linked to the feedstock itself. So what becomes crucial is the feedstock pretreatment because of the recalcitrant it poses, especially agricultural residues for that sense. And we can broadly classify the pretreatment requirements into 2 categories. It is either microbiological requirements or engineering requirements, and the categories of pretreatment itself could be 4 different categories. So the microbiological requirements mainly are that we need to ensure there is enough hydrolysis happening or enough microbial activity happening on the feedstock itself to pass it through different stages of AD. For that to happen, we need a good enough particle size of the biomass and increased surface area so enzymes can act upon easily. And a pretreatment step should possibly give us good sugar release. For biomass, it is not that critical for sugar release because the complex microbiology, which is synergistic in nature, will take care of hydrolyzing the cellulose and hemicellulose. So sugar release is not that critical for the biogas side of things. But what is important is not to lose the polysaccharides. So a pretreatment step should ensure that there's no loss or no inhibitors are being formed. With this being the microbiological constraints, the engineering constraints, again, we've been talking about this since morning. It needs to be cost effective, scalable, and energy efficient with all 3 linked. So for a pretreatment process for biogas production, what we generally expect is one of those curves that we see. So the black curve there is without pretreatment. When we feed the digester with the feedstock, we end up producing biogas. But then what we would want to see with pretreatment is either an increased rate, which is the blue curve that you see, without increasing the final yield of it, or we would want to see an increase both in yield and rate of biogas. And in some cases, what we will also notice is a short lag followed by an increase in rate and yield. What this means is that the pretreatment process has possibly induced some inhibitors in solution, which is possibly inhibiting the microbial growth. So there's a lag time where the microbes will actually acclimatize to the new feedstock type and then start producing the biogas. But what we definitely don't want is this red curve here, where there is a big lag and there is a significant drop in yield. What this means is the feedstock pretreatment has gone beyond the optimum level and we've lost enough organics in the feedstock. And there's also inhibitors in solution, which is why we see an inhibition stage here, followed by a biogas production. So pretreatment techniques are, again, 4 categories. We know it's physical, chemical, biological, or physical chemical. With physical treatment, what we mean is just reducing the particle size, basically. And the particle size reduction is affected by 2 important parameters, one being the moisture content of the feedstock, and 2 being the ratio of the initial particle size to the final particle size. When that increases and the moisture content increases, the pretreatment energy goes up for physical pretreatment specifically. So we don't need to really focus on that too much if the feedstock is going to be really high in moisture content. Next, moving on to chemical pretreatment methods. For example, if we take an alkali pretreatment method, considering that alkali acts on the lignin fraction of the biomass, it can be solubilized, and then we will end up with predominantly cellulose rich with some hemicellulose in the biomass, which could be better for further processing in terms of AD, because when the cellulose fibers are exposed, it's easier for the microorganisms to hydrolyze it. Similarly, in biological pretreatment, we've got enzymes acting upon. But again, what happens in these two senses is that with alkali pretreatment or chemical pretreatment, we would need a neutralization step prior digestion so that it can favor the biogas production yields and rates. Similarly, for biological treatments, it could be quite time consuming, and it could be quite slow, and it is also expensive to recover the enzymes. So at least with the biogas production, it is not that common to employ enzymatic techniques as pretreatment methods. What is mostly growing these days is the use of physicochemical pretreatment, such as steam explosion or cavitation, which is what I'm going to talk about. And what happens in this treatment is it utilizes the factors from physical pretreatment and chemical pretreatment, combines it, and then pretreats the biomass much more efficiently. So what we see there is some biomass fibers, which is just floating before pretreatment, and upon pretreatment, we can see that the particle size has gone down, as well as the solutions become turbid, meaning there's some chemical products in there as well. So what is cavitation? I think everyone would have heard about ultrasonication. Ultrasonication is a form of cavitation. Cavitation in general is the phenomenon of formation, growth, and collapse of vaporous bubbles. When the local pressure in the liquid drops below the vapor pressure of water, what happens is vaporous bubbles tend to form. And with the pressure becoming more negative, the bubbles tend to grow and then finally collapse. This collapse will induce a shear and high-speed jets, which acts upon the biomass, physically pretreating the biomass by breaking it down and modifying the surface morphology. Whereas the hotspots created will have high extreme temperatures and pressures locally. These can actually cleave the water molecules to produce reactive oxygen species, which would then act upon the biomass chemically to partially hydrolyze them. So there's no chemicals involved in inducing cavitation. Cavitation is another common term that engineers come across, and they don't want to come across as well, because cavitation is quite damaging. And there is a separate body of work that's going along the sidelines or parallelly to avoid cavitation in propellers and pumps, but controlled cavitation can be harnessed for great benefits. So cavitation is of 4 types: optical, particle, acoustic, and hydrodynamic. I would not be talking about optical or particle because it's not of relevance to biomass pretreatment, whereas acoustic and hydrodynamic are. Acoustic cavitation is nothing but realizing the effects of cavitation using ultrasound. With ultrasound, it is mostly in a horn-based system or a bath-based system, so it's not really scalable. It is energy intensive. It's got its niche applications like surface cleaning and medical applications, but for biomass pretreatment, it is unlikely to employ acoustic cavitation on a higher scale to do this. As an example, you can see there on the left-hand side, there's a grass strand that has been that is before pretreatment, and upon sonication for 20 minutes, we can see the fibers opening up in the middle figure. And the last one, the fibers are completely shattered, and we can see that there's a solution with some small fibers and some chemicals in there, of course, you can't see. It might be noisy, but just bear with me. Yeah, so the horn acting upon the grass strand there. So this is a typical ultrasound horn that is acting on a biomass particle. And we can see the processing volumes are restricted here in this case. And depending upon the type of horn and size of the horn, as well as the frequency, we can't go above a few liters. So for that sense, hydrodynamic cavitation is preferred. And hydrodynamic cavitation is generally realized by using a constriction in a flow path. And what happens when having a constriction in a flow path is the cross-sectional area reduces, which means according to Bernoulli's principle, we tend to get down to a very low pressure in the local region with a very high velocity of the flowing fluid. And this induces cavitation bubbles in the flowing stream. And these cavitation bubbles can then act upon the biomass both physically and chemically. Generally, hydrodynamic cavitation can be produced using 2 types of devices, either rotational devices or one without moving parts. Rotational devices are rotostator, which I haven't actually shown here, which I should have, but it's got some maintenance issues, and it's high energy consuming devices, so it can't be used efficiently on a longer run for large scale applications. Linear flow devices or stationary devices for that case are again of two types. We have linear flow devices and rotational flow devices. What I've shown here in the graph, the picture here is the top one is a orifice plate and the bottom one is a venturi tube. So it's got either a small constriction or a nozzle that can actually generate cavitation. And if we're talking about agricultural residues as feedstock that needs to be pre-treated using cavitation, we can't use these devices because they tend to clog. So a pre-milling step becomes mandatory. And because pre-milling is energy intensive, we don't want to go towards using these devices. And another major disadvantage of this device is the cavitation bubbles are formed along the walls of the device, which means the cavitation bubbles, when they collapse, can erode the device itself, leading to further maintenance and other problems at scale. So what we are trying to do is use a vortex-based aerodynamic cavitation device, which employs swirl flow. And what happens in this particular device is there's a tangential inlet and an axial outlet. The tangential inlet has no small constriction, so it's a proper big hole, unlike the linear flow devices. And what happens here is when there's a flow of the feedstock slurry happening through tangential flow, it sets up a free vortex in the circular chamber. And then through the axial port, because of the forced vortex, it needs to force out of the chamber, and we end up producing cavitation in the core of the flowing liquid. What this means is because the cavitation bubbles are formed in the core of the flowing liquid, the device by itself is protected. So again, there's no small holes, which means it is not prone to clogging. And we've got an early inception of cavitation, meaning there's less energy consumption compared to the state-of-art devices that we've just seen before. And low energy consumption obviously means low operating costs. And this will be favorable for an industrial application. It can be retrofitted on and because of low footprint as well. I'm going to just talk about one of the projects that we are currently on. So this is a biorefinery project that we are currently working on. Is an Indo-UK project. And it's a consortium with both industries and in academia, both in UK and India. And we are the lead partners on this one. If anyone's interested, please visit the website there. What we're trying to do is utilize the sugar industry waste and then produce value-added products and biofuels in a biorefinery approach. For anyone that's not familiar with how the sugar industry works, so sugarcane, which is the predominant source of sugar in India, is harvested from fields, and then the top of the sugarcane is cut off, the leaves basically, and they're called sugarcane trash, which is one of the most important feedstocks. And again, once the sugarcane trash is cut off, the stems are transported to the industry where it is crushed, and upon juice extraction, we end up producing molasses, which will then be fermented to produce spent wash. It is also one of the most significant wastewater streams in the Indian context. Again, during the process of sugar production and the logistics of it, we end up also producing bagasse, which is the crushed stem, and then press mud, which is the clarified mud plus some fibers. So these are important feedstocks that we're concentrating on in this particular stream of work. And I'm going to just present results on what happened with cavitation of bagasse and spent wash in the next few slides. Again, as you can see, the quantities are immense. It's a few million tons of, a few hundred million tons of bagasse, and spent wash is in billions of liters. Bagasse, what happens is it's got about 40-45% cellulose, about 20%, 20-25% of hemicellulose, and 25% of lignin as well there. So we don't want to be concentrating too much on deconstructing the biomass, which could actually be a strong word. So I should probably say fractionation of biomass. We don't want to go towards that side, but because we are focusing on biogas, we want to enhance the rates and yield of biogas generation. So we would want to probably improve the efficiency of the microbiological process. So for that case, we would want to see where the optimum pretreatment stands. So instead of solubilizing fractions of biomass, we are trying to basically improve the physical properties of the biomass in terms of improving surface area and the morphology of it so that the microbes can act upon better to improve the rates. So upon the untreated biomass and 9 passes treated biomass show very minimal difference. And this is expected because for a very low treatment time, which corresponds to number of passes, you will not be seeing as much of a chemical change in the biomass itself. But in this particular study, we've done over 100 passes, and we did see the shift in patterns in the FTAR profile that I've not shown actually. What we also noticed with a longer number of passes or a higher number of passes is that hemicellulose tends to solubilize. And then we're left with a better biomass with high purity cellulose. So we don't want to go towards that, because for biogas generation, it is good enough to pump in the biomass as crude as possible. And that will actually keep the costs at bay. So we don't have to spend too much energy or money trying to pretreat the biomass. So here 9 passes was found to be optimum. And we can see it induced about 24% increase in biomethane generation. And what we could also see is the lag phase is virtually disappeared. And what this means is the complexity of the feedstock is reduced. And if the complexity of the feedstock is reduced, that tends to increase the rate and yield of the biogas generation. So I should mention that this work was a preliminary work. So we used milled biomass. So what we've done next is to use the as received bagasse itself. So these bagasse particles are as long as our fingers. So it's quite long fibrous materials. And untreated biomass, as you can see in the leftmost figure, is all big long fibers. And upon pretreatment, the particle size tends to go down. Of course, I'm not showing the chemical profile here of the liquid phase products, but then we also ended up producing some acids as well as C5 and C6 sugars. In terms of energetics, what we did observe was, compared to acoustic cavitation or ultrasonication or physical methods, which could be very high in terms of energy consumption, a few thousand kilowatt hours per ton of feedstock, the vortex-based device was actually quite low in terms of energy consumption. So at lab scale, we operated there about 140 kilowatt hour per ton for what we used, which was about 10 kilogram per meter cube loading. And with industrial scale being in this range of 100 to 150 kilograms per meter cube, we can see that the energy required keeps going down, assuming there is a steady or a stable increase in terms of energy gained from the enhanced biogas generation that we see. So the margin keeps going up with increase in the feedstock loading, and which is a good sign, because we know that at lab scale, we could still do it at an energy efficient manner. So at industrial scale, it's going to be better. So what we did next was we tried using spent wash as a feedstock, and then we performed the same kind of a pre-treatment. And we could see that with spent wash, it is about 2 passes, which was quite good enough to increase the biogas yield by 14%. Also, what we see here in biogas generation with spent wash is that there is a small hump in the beginning. What this means is that there are some residual sugars from the wastewater that is being used up by the microbes. And again, as I said previously, the chemical effects are not as much in the lower number of passes regime. So we don't see a loss of smaller sugars upon cavitation pretreatment as well. So the untreated and pretreated biomass here quite overlaps nicely, which means there's no loss of small sugars in 2 passes pretreatment. And upon that pretreatment, what we do see here is the lag time being considerably low. So there's about 1 day that we've gained in terms of lower lag time with pretreatment. And then what we also noticed was a smooth proper profile of biogas generation instead of a dioxic phase that we see in untreated biomass. So what this dioxic phase means is that because it's complex COD, we've got melanoidins, and then we've got other compounds that are quite difficult to break down. With cavitation pretreatment, we could actually break down these complex COD to usable bioavailable COD. And this has actually resulted in an increase both in rate and yield of biogas, as well as reduced lag time. Again, what we also wanted to see was a 1 and 2 pass pretreatment effects. Again, the energy keeps going up when we increase the number of passes. So we tried with 2 passes and 1 passes here as well. And we can see that with 1 passes pretreatment, the energy required is lower than 2 passes, assuming that for both cases, we get the same energy output or enhanced energy output.
Sanjay?
Yes.
I'm sorry to interrupt, but you're 5 minutes over. So could you wrap it up?
Yes, it's just last 2 slides, I'm done.
Thank you. Appreciate it.
So what we see next in this graph on the right-hand side is our scale-up data. So we work with a lot of industries in India as well. So in central India and Western India, we managed to scale up this device to a proper higher industrial flow rates, as we can see, from 10 to 50 meter cube per hour. And then we could get an analogous increase in biogas yield similar to what we observed in the lab scale. So this being a biorefinery project, what we're trying to do next is utilizing digestate, because we still have some unconverted feedstock in digestate. which is rich in cellulose. So we want to use that cellulose as feedstock towards other value-added chemical production. And finally, what I would want to summarize is AD is an excellent technology that we could actually harness to valorize biomass. It could also be a good biorefinery base, provided that the pretreatment technology is energetically and cost effective. And thinking beyond biogas, biorefineries is the way forward. Of course, we all know that. And I would like to finally acknowledge the funding support, and thank you very much for your attention, and sorry for going over time.
Thank you, Sanjay. Nice quote as well there on your last slide. Really interesting to see the engineering solutions that you've proposed for those. So if anyone's got any questions for Sanjay, please can you put them into the chat? And we'll move straight on to the one and only Scott Banks now. Scott now, who's going to talk to us about solid oxide cells and biogas. John, are you ready to go?
Yes, can you hear me?
Just about.
Okay, sorry.
You might need to speak up a little bit. Okay, we can see your screen now.
Okay. Okay, is that clear?
It is, yeah. Away you go. Thank you.
So this is a little bit different. We come more from the hydrogen and energy materials community, but bioenergy is really important in catalysis in what we do. So if I just say a little bit briefly about things that we look at in St Andrews. So from the materials perspective, we're looking at converting oils on the top left. We're looking at lignocelluloses using pyrolysis and photocatalysis. We're also interested in biochar. And I guess from the fuel cell point of view, syngas, or electrolysis point of view, syngas is very interesting to us. So today I'm going to talk about biogas, which comes from organic waste. We already had a couple of presentations that are related to that. And in particular, at the end, we'll talk about electrolysis. So initially about biogas reforming, this is what happens at the electrode or before the electrodes. Some work on fuel cell testing of biogas, and then some more recent work on biogas upgrading. So I guess you all know, but what is biogas? It's a gas that's produced from organic waste. So it can be from plants, from ryegrass silage or manure or whey or waste bakery products. It can be from animal manure, it can be from sewage sludge, and in the urban environment, there's a lot of waste, organic waste from households that could be used. And what's in biogas is what, from fuel cells, we talk about it being a very good dry reforming mix. So about 60% methane and 40% carbon dioxide, a little bit of water vapor, not much else. But important amongst the not much else is some hydrogen sulfide, which can be from tens to thousands of ppm. And that's the one thing that can impact catalysis and electric catalysis. So from a project we had doing biogas in India, we visited a small dairy farm in West Bengal. And this is it's a cooperative with a dairy farm producing biogas. And there's a lot of biogas in India and the developing world. And the thing to note was that the Lister engine, which looks like is a 1930s shearing engine to produce electricity. So you go to the trouble of producing the biogas, and then you convert it at maybe 10% efficiency of energy. And that's something that we think if you can combine fuel cells and biogas, you can get twice as much electrical energy from the same inputs. And so looking at that in schematic, you have the anaerobic digester. It produces methane and CO2. which you can put into a combustion engine, but we prefer into a fuel cell, and that gives us electricity. So in electrochemical system, you have heat and electricity, and you've got methane, CO2, hydrogen, carbon monoxide, and water. And then if you use electrolysis, you can increase the amount of reducing gases. So particularly the carbon monoxide and hydrogen will increase. And then that can go into a methonator to produce synthetic natural gas. Or something that I think is really important is you can produce a gas that has got suitable flammability to feed directly into natural gas grid. So you could feed this sort of biogas into a gas grid along with green hydrogen, and you could have a new natural gas mix of similar flammability and combustion properties to current natural gas. So whilst the input is mainly CO2 and methane, of course, when you heat it up into a fuel cell or electrolyzer, it reforms. So it's an ideal dry reforming composition. So obviously the methane content decreases, and we reduce carbon monoxide and hydrogen as well from that. And the point that we worry quite a lot about is the coking point. So if your methane containing mixture decomposes to yield carbon, that destroys the catalyst, and you need to add some oxygen or some steam into the system. CO2 as dry reforming is quite good for that, but you can see that about 720 and above, the equilibrium is there's no coking, and it doesn't really matter you have CO2 and methane to start with, or you've got reformed CO2 and methane. The carbon, hydrogen, oxygen ratios are the same. So the thermodynamic point for carbon deposition is the same. Obviously, the kinetics you may play tricks with kinetics, but essentially the higher the temperature, the less problems you have, and also the less more oxygen. So looking at the gas mixes from equilibrium, you can see at 720 you'll have no carbon decomposition, and essentially you've got mainly hydrogen, carbon monoxide, and some methane at equilibrium. An example of a steam reforming catalyst is this X-solution rhodium nickel dope titanate, and that works quite well for reforming methane and CO2 into carbon monoxide and hydrogen. We fed 5% hydrogen sulfide into that, and its activity died, which is not a great thing. But the more important thing is that when you took the hydrogen sulfide away, the catalyst recovered quite quickly. And often that's what you need, because you need to scrub the sulfur from the inlet, because if you don't scrub it from the inlet, it'll be in the outlets, and you can't really put out sulfur dioxide either. So you need to remove sulfur somewhere, but you need to have catalysts and fuel and electric catalysts that don't die when they see sulfur. So looking at an example of a fuel cell system, this is a nickel-based anode, nickel zirconia, and we infiltrated a small amount of a proton conductor, barium, zirconium, cerium, ytterbium oxide, and that's known to prevent carbon deposition, and that's about 0.6% and you can see it on the bottom right of the infiltrated or impregnated proton conductor. That gives you pretty high performance, and you can see that the three-stage impregnation, the 0.5%, gives you the very highest performance. And 2 amps per square centimeter at 0.8 volts is a really good performance. It's as good or better than you get on pure hydrogen on a similar gas mix. So you certainly can develop fuel cells that work well in biogas, and this also has some sulfur tolerance as well. In terms of here, you can see that when you change the flow rate, you start to have limitations in the power, and in the top left, you see that the voltage is dropping as you draw more current if you don't have a high enough flow rate. So that means that you're starving the electrode of fuel, which means that you're having a very high conversion, which is a good thing. And then running it for 50 hours in the lab gives us pretty stable current density. These are kind of running at the limit of the current we can cope with in the lab at the moment. So then if I move on to a very similar system, but this time driving the current in the opposite direction. So this is upgrading biogas through CO2 electrolysis. And this work was performed with, or supported by LERG, a company in Dundee now, interested in electrocatalytic reactors. So the sort of reaction, if you're removing CO2, you'd expect CO2 to go to carbon monoxide, steam to go to hydrogen, and the counter electrode, you'd be taking oxygen away from the overall system. But there are side chemical reactions to that, and in fact, the electrodes are quite catalytically active, so you get a lot of dry reforming. So you don't, it's not CO2 that you're mainly working with. It's the reformation products. You have, or sorry, not methane. It's the methane is reformed. You also have reverse water gas shift, so CO2 and hydrogen will reform to CO and steam. You'll have methane steam reforming. You could have methane cracking, which we try to avoid, and also you can get carbon from the Boudard reaction. So there's a whole series of complicated reactions, and you're trying to effectively electrolyze the CO2 content and leave the methane content intact. So this experiment was performed on a nickel-based electrode with lanthanum strontium manganite air electrode. You can see the electrolyte there, and looking at 3 different gas mixes. This is methane, CO2, and the more carbon dioxide, the higher the current, because you're in electrolysis. So the carbon dioxide is your fuel that you're converting, and at 1.4 volts, that gives a pretty good performance and works better in the high CO2. In the lower CO2, you're starting to run risk of a little bit of carbon deposition, which is part of the degradation. So when we look at the gas outputs, you can see that there was no carbon monoxide to start with, but at open circuit voltage, the first thing you see is 30% carbon monoxide. So the gas has reformed towards equilibrium. It's not at equilibrium, but the overall calorific value is not going to change that much due to the reforming, because you're just reorganizing the gases. But then when you do electrolysis, you increase the carbon monoxide content, you increase the hydrogen content, and you reduce the CO2 and steam contents. So you end up with a gas that becomes higher calorific value, and you don't need to change it that much to make it much more miscible with natural gas. And the higher the voltage, the higher the current, and the more conversion you get. So looking at the gases overall, this is just picking one composition, one mixture. So as you can see it better, the CO goes up. The hydrogen seems to stay much the same, so the equilibrium readjust around the hydrogen. Methane slightly increase, so you've got 20% methane, 26% hydrogen, 38% of carbon monoxide to start with. And effectively, we are actually taking the CO2 out, but in a very indirect way. So the CO2 content is decreasing as we do more electrolysis. And it's perhaps clearer if you look at the difference in concentration. You can then see that the CO2 is decreasing as the current increases. So as we go up to 1.6 amps, we get quite a significant decrease in the carbon dioxide content. So yeah, so you have three processes going on: the CO2 electrolysis, the steam electrolysis, and then also the water gas shifts, which then can adjust what's being reduced. You don't know exactly what you're reducing because it's a very complex dynamic system. So as we come to conclusion, we can get better. It's easier to reduce with more CO2s, although your starting gas is less useful. Methane and CO2 are converted to hydrogen and carbon monoxide under OCV through normal reforming. Cell performance is more stable with lower methane and inlet gas. And we're now looking at an alternative electrode, which is going to be a little bit more stable to methane concentration. And just some preliminary results on nickel doped lanthanum calcium titanate. We've used this for electrolysis before, and you see, if you look at the top right, you see we compare a graph in black and a graph in green. The black is the nickel electrode, which degrades. So this is at 60% methane, 40% CO2, which is probably the level you'd like to be starting with. And there's some coking problems, I think, with the nickel. With the exalt perovskite, that seems much more stable, and that's where we're looking at the moment. And you can see at the bottom the exalt nanoparticles of the titanate on the left and the right. And just finally to thank LERG or LERC, their US parent. I should mention the hydrogen fuel cell SuperGen hub, and we're very keen that we can link with the Catalysis activities and especially the bioenergy hub. I think that's really important going forward. And Nushi did the electrolysis work. Stephen and especially Janjun did the fuel cell work. So thank you.
Thank you very much, John. Really interesting and something a little bit different to hear about today. So there are no questions on the group message board. So I'm going to move on now to our final presentation of the day, which is from Andrew from Manchester. He's going to talk to you a bit more about the work going on within SuperGen about the systems and vectors. So Andrew, over to you.
Thank you, Katie. Can you hear me okay? So yes, thanks for inviting me to speak today. I'm from the University of Manchester, and I'm going to introduce to you what we're doing in SuperGen in the vectors and the systems themes. So moving straight onto it. I think you might have seen this diagram earlier today already. So basically the vectors theme is led by Marcel McManus at the University of Bath, and the systems theme by Miriam Roda at Aston University. And essentially we are part of the wider SuperGen Bioenergy Hub. The vectors theme focuses on identifying preferred bioenergy pathways that will produce basically the appropriate bioenergy vectors that the UK needs, analysing the role of different bioenergy vectors and how they can work within a wider UK energy network. And the vectors theme also determines how these may fit together to complement the bioenergy, bioeconomy and carbon reduction strategy. Most of my work takes place within the systems theme, which is focused on identifying and evaluating the impacts of bioenergy sector. We really want to maximize the benefits of bioenergy through reducing emissions, reducing reliance on fossil fuels, and where possible, finding some wider ecosystem benefits that may be achieved. And as we scale up bioenergy technologies, much of our work focuses on looking at what the impacts may be as a result of that scaling up, and where we can find complementary activities with other sectors. So in a nutshell, this is what we do. We basically look at bioenergy systems in the round. We interrogate them, we map them, we work out right from the start where feedstocks or resources are grown or mobilized, what happens to them, what they process, where they source from, what transport takes place, what fuels are generated, and ultimately what kind of bioenergy vector is produced at the end. So we interrogate different bioenergy systems, and for all of these, we benchmark them and compare them through doing things such as looking at process energy demands, analysing the GHG performances, looking at water footprint, what kind of particulate emissions or other pollutants are produced, what kind of products are used along the way, what are we competing with, what resources are we providing that other sectors could be used, of the emissions that are generated, what are the temporal emissions dynamics, as in how long do those emissions stay up in the atmosphere. Finally, we look at things such as the techno-economics. What's the costs, and is it cost effective? Is it possible to do these things? So we kind of take a step back, look at the whole system of bioenergy technologies, and kind of scrutinize what's going on and trying to make it a viable system to achieve each of the UK's targets. So going back to the beginning, we need to, as a sector, basically ensure that the bioenergy systems we're working on are designed to complement and to meet the UK's energy demands. You may be familiar with these graphs, some of you may not be, but I think that people are not aware that bioenergy almost already has a clean sweep of its contribution. So if you look at the top diagram, top left, UK energy, from the UK's renewable energy contribution, bioenergy already contributes the largest fuel to that mix. For renewable electricity, bioenergy is the largest contributor, things such as pellets and wood fuels, providing a lion's share of that. Renewable heat, bioenergy is also leading the way compared to other renewable technologies. Finally, renewable transport fuels, bioenergy or biofuels are the predominant sources of our renewable transport fuels in the UK, bioethanol and biodiesel, with all the work taking place looking at things such as aviation fuel. So bioenergy is already there, very much on the agenda for the UK, and the UK government are targeting it to be an increasing force to provide energy, jobs, low carbon emissions, all the rest of it. So bioenergy here to stay, and it's our job to make sure that we do it in a sustainable way. What does this mean? How does the bioenergy fit into the bioeconomy? Well, the bioeconomy in the UK is massive, 220 billion according to their output, with 5.2 million jobs currently focused around the bioeconomy. By 2035, the UK think that biotechnology and bioenergy sector may contribute £8.6 billion worth of money. So there's lots of money focused on this bioeconomy, and the UK government are really pushing for it. But the final statement is something that us as a sector need to focus on. So for a healthy bioeconomy is really going to need an abundant, appropriate feedstocks that are efficiently used. So it's up to us as a bioenergy community of researchers to ensure that we're using the resources efficiently, we're generating the energy that the UK needs, and we're basically complementing our research with what the UK needs to develop this bioeconomy and to achieve those large turnover annual rates. From an emissions perspective, many of you know that we're doing bioenergy because of emissions. I used the CCC, Committee on Climate Change, as an example here to demonstrate this. So they acknowledge that biomass can be produced and used in ways that are both low carbon and sustainable. Great. And they also acknowledge that biomass can make a significant contribution to tackling climate change. Another great. But the risk they identify is that there are risks that biomass production and its use can in some circumstances be worse than fossil fuels. So again, this is something that we focus a lot on the systems theme and the vectors theme, and something that the whole bioenergy community should be aware of and focusing their research to ensure that we know what the risks are, and at every stage of reducing emissions to try and increase the emission profile of bioenergy systems. I've shown this graph quite a lot, or this diagram quite a lot over the years, so I won't go into much detail, but the take-home messages are essentially between now and 2050 and beyond, the focus on bioenergy systems is going to be growing in the UK. That means we need more resources. We need to focus on developing technologies, but there's also going to be a lot of dynamics taking place there. The amount of resource required by that bioenergy sector is going to increase, but it's also going to change. Different types of vectors, heat, power, biofuels, are going to require different fuels over the timeline. As different technologies come online or different technologies fade away, there's going to be lots of dynamics there. So we need to be nimble of knowing what resources may be available in the future and making best use of those where possible. So the Vectors team are currently doing some work around looking at these dynamics about mapping into the future, what resources may be available, what resources may be available for the UK, and looking at how best to use those to balance our UK bioenergy demands. In the systems team, we've recently done some work around models. Models are used very heavily by decision makers and the policy and government to basically inform their decisions. They model all sorts of things to do with bioenergy to work out how in the future, what can they use in the future from a bioenergy perspective to meet their demands and balance their targets. So our assessment, without going into massive details, essentially we looked at different types of models, and depending on what model you use, you get different results. Different models are better at covering different scales of issues or different dimensions of issues. But the take-home message is you need to be really aware of what model you're using or what that model is good at and what it's not good at. And a piece of spin-off work we've done for the Department for Transport recently is some work for them, basically highlighting how much resource may be available for them for the future biofuels in the UK, and are the models we have in the UK fit for purpose for basically allowing them to form their decisions or not? Are they aware of all the interactions and nuances of bioenergy systems, and what kind of new models may be required to fill in some gaps? A large part of what we do in the systems is making sure that bioenergy becomes a low carbon renewable energy option. And to do that, as I said before, we go right back to the beginning. We look at every step within a life cycle, from the production, the processing, the harvesting, the transport, all the way through to combustion. It's important to work out what emissions are taking place, what energy is going in, what are the fluxes between CO2, between the different systems and the atmosphere. And allowing us to do that, we do lots of LCA analysis, where we analyze bioenergy's GHG intensity. So this is a random graph I plucked out from some work we did a few years ago, looking at how we can generate low carbon heat and how that heat compares to natural gas or other fossil fuel technologies, as in, can bioenergy provide an alternative solution to providing heat with lower carbon intensities compared to fossil fuels? And the answer is yes, it can, but all those dots all over the place demonstrate that there are examples where it does not. So it's important to investigate each supply chain intently and work out what the risks are and what we should and shouldn't be doing to maximize our GHG performances. The secret to emissions performance is all about that flux of emissions and the balance between CO2 coming from the atmosphere and going back to the atmosphere. It's really important to have a favorable balance for bioenergy to be a good option for the UK and for everyone. So essentially, we do lots of work measuring measuring the whole system and working out where the emission flows are. And essentially, we want to ensure that bioenergy results in a no more net flux movements of CO2 to the atmosphere compared to fossil fuels, if it's going to be a viable energy technology to replace those. Here's an example here, some work we've recently done with SuperGen, investigating this emissions issue. We produced 2 policy briefs, these which are available on our website's downloads. They investigate and provide some really clear guidance on emissions. The first one focusing on timing and temporality of emissions, as in when you do different bioenergy technologies, what emissions do you generate, what specific GHGs, how long do those GHGs spend in the atmosphere, and what does that mean for our climate change targets? It's something people don't really think about. Different systems produce different types of GHGs, and depending on what they are, has different impacts long-term on the climate change issue. The second policy briefs were led by the systems team, focusing on how these emissions are accounted. Emissions are accounted through the UNFCCC emission accounting framework. This is important because it allows you to account emissions, and this is what's used by the climate change people to work out whether emissions inventories are going down or up, and what the contribution of bioenergy would be to reducing emissions in those inventories. So if you want some clear guidance on either temporality or the emissions accounting of bioenergy systems, please have a look on the SuperGen website, and there's some clear guidance there developed for lay audiences. Finally, sustainability is another big issue we're looking at. Sustainability bioenergy being a strange thing because there's so many different indicators of what sustainability is. Things ranging from soil quality, energy diversity, water quality, many sustainability indicators that cover bioenergy systems. The graph on the right is something produced by Paul Gilbert and Patricia Thornley a number of years ago, and it clearly demonstrates that this given bioenergy system is sustainable, but in other aspects is not sustainable. So sustainability for bioenergy is very much a trade-off between these different indicators. So it's up to policymakers and whoever's making the decision to work out what they want to prioritize and what sustainability actually means. To help with that in a minute, we developed a new model, which is currently in the development at SuperGen. We called it very cleverly the SuperGen Sustainability Indicator Model. And the aim of our model is to provide a research tool that enables assessment and comparison of sustainability of different bioenergy systems, looking at feedstocks, technologies, and supply chains. And we essentially want to provide a tool that allows us to analyze any system, bioenergy system, and analyze it and compare it one against another to analyze what sustainability is based on a series of credentials. And interestingly, we're focusing on the SuperGen case studies, one of which is very much Catalysis focused. So there is work going on at the minute to analyze the sustainability of Catalysis. This is a diagram, which I'm gonna very quickly go through. This is our model. So basically we look at the whole system, bioenergy system, from the start, from the feedstock through to the conversion to energy technologies. We assign different indicators along the supply chain, indicators focused on people, focused on development, focused on natural systems, focused on climate change, and what indicators are relevant to the system. We then develop a framework of sustainability. These are the indicators that are relevant to the system. And then we do analysis, analyzing whether a different indicator provides a negative or a positive impact. And if that impact takes place, what is the scale of impact that may be? And there's some clever maths taking place. And for each bioenergy system, we develop a sustainability score, which is index value, to try and benchmark and our comparison of different bioenergy systems one to another, depending on a long list of sustainability indicators. This graph here is basically a screenshot of the model itself. And as you can see, without going into much detail, there's red bits and there's green bits. So for development, there's certain areas where there may be an impact and there's certain areas where there may be a positive impact. So positive and negative is all a balance, but it's a clever tool that allows you to visually analyze the sustainability of a supply chain or bioenergy system and to work out what sustainability is. So if you would like to know more information about that, please get in touch. It's currently being developed. My final slide focuses on the SuperGen UK Bioenergy app, which is currently taking place. We're sending out lots of emails. You may have heard of this, you may not. So if you want to get involved in developing this, please send me an email. At the very least, we have a encyclopedia page or feature within the app, which will allow users to work out what all the jargon words are, what is bioenergy, etc., etc. So if you'd like to contribute to that or contribute your research and get noted, we are currently developing that. So please send me an email and get involved. That was a really whiz through what we do with the vectors and the systems theme, but I'm happy to answer any questions. Thank you for having me here today.
Thank you, Andrew. That was great. And I think it's really important for everyone to remember that when you do your research at small fundamental scale, that then it does have to fit into these larger scale systems and does eventually have to be implemented. So the systems and vectors work is really important to see how it does actually fit into the real world. And then all of those keywords that Andrew mentions that everyone's really interested in, like sustainability, greenhouse gas emissions, that all happens through the work that Andrew does. I've had an advanced preview of that sustainability model, and it is really, really good. So Andrew, when will that be released to the public?
We're still working on it now. We're hopefully, definitely by the end of the year, at the very, very latest. It'll be in the next few months. There'll be something available on the SuperGen website, first and foremost. That's
great. And for the app, when do you think you'll be releasing that?
The app's currently being developed by over the summer now. So it's actually, right now it's being developed. So if you'd like to get involved, please email me. The app will be available from October onwards, again from the SuperGen website.
Yeah, and that's another really good way of, we're going to use it as an education tool as well. So it's going to be a really good way that we can help people understand what's going on in the world of bioenergy. So please, I encourage you to get involved with that work. So are there any questions for Andrew? I'm just going to have a look on the chat. Okay, no, there's no questions at the minute, Andrew, but so thank you very much for your presentation.
Thanks, Katie.
And I'll speak to you soon. Okay, everybody, so we've got some time for discussion now, so I'm going to invite Chris and Patricia to jump back in if they're still around.
Yep.
And while they're jumping back on, I've got a question for the audience. So we've seen that there is a lot of potential for work between the Bioenergy Hub and the Catalysis Hub. But what do you think are those hot topics that we should be looking at projects on? So is it plastics? Is it looking at waste? Is it looking at upgrading? So from our audience, I'd like to get a general feel of what are those topics that you'd like to see the two hubs working on. And while people are typing away, Patricia, what are your thoughts on this?
I was really interested in the work earlier on synthesis of plastics. I think it's something that there's a whole host of issues around, because you've got the specification of what you're actually replacing. And there was some work done a few years ago by, I think it was LBNet, on what products we should be replacing. And it's important to understand the carbon intensity of the product that you are replacing in order to prioritize the right ones. But also it's about the process that you go through. And if, you know, the process from bio requires a huge amount of energy to be input, then there is no point. We might as well have used a fossil fuel derivative in the first place. So I think that people tend to intuitively think that because bio is a good idea with efficient, sustainable processes in comparison to fossil fuel liquids, electricity and gases, people intuitively tend to think the same is true when we move to polymers and plastics. And I think the evidence base around that is really quite poor. So I would like to see some closer collaboration on that, where we look at the actual processes that are used and the products that we are displacing, and actually crunch the numbers to prove that it's a good idea.
Yep, that's great. Patricia, I agree with you completely on that plastics issue. It looks like a really interesting one, and there isn't a lot of information out there. So that's a really juicy one that we could get working on. So Chris, what are your thoughts on this?
Yes, I think the plastics one is a good one. I think also hydrogen production is the other one. So we had a talk from Mike earlier on. We are also working on hydrogen, both from plastics and from biomass within the SuperGen hub. There's stuff that that John was presenting in terms of syngas and so on. So I think there are a number of hydrogen vector questions that we should look at. And actually, it does go back into the plastics as well, you know, because we can also do the reforming of those plastics too. So I think there is something in the whole area of hydrogen as well as the plastics, I think.
Yeah, and hydrogen is just a really hot topic at the moment as well. So what are the other hot topics, would you say, in this area?
So can I just comment on the hydrogen one a moment, Katie, just to let people know. The Bioenergy Hub had a meeting earlier on this week with the Renewable Energy Association, and we will put this out in our next newsletter, but they're hosting their annual green gas event in October, which is also moved to an online forum. And there's a lot of interest there around the specification for alternative gases and the purity of hydrogen that's actually needed for different purposes. And really, I think there's a need to raise awareness, you know, like the presentation this morning focused on, about the fact that there are pathways to hydrogen other than electrolysis. You can actually produce it directly. So I'm hoping that we will have some form of slot or collaboration with the industrial side at that event in October. So if you sign up with SuperGen, we will make sure that you get told about that, and we'll try to synthesize maybe something from across the bioenergy and catalysis communities for that. There is similarly a government initiative on at the moment trying to collate information on priority areas for national research needs. And one of the ones that, again, we're going to be flagging up there is this need to think about the standards and testing and quality of alternative liquids and alternative gases. So the idea that there might be a need for some national fuel standards and testing agency or something. I don't like the word agency, sounds like American CIA type stuff, but you know what I mean.
Yeah, I know what you mean. Okay, so I'm just looking at the chat now, and no one's got any. Oh, here we go. We've got something about air pollution. Making sure that when developing these processes, the additional need to reduce the PM10s and PM2.5 levels, and not just the embodied energy and CO2 of products. Yeah, I think air pollution is a critical issue, especially when you look at the legislation that's been put into place now around air pollution, and a lot of cities looking about making their cities clean air zones, which is all about reducing those particulate levels. So, and that's something that should be considered as part of the sort of systems vectors type of activities, looking at do these processes actually have a beneficial impact on our environment from all aspects, not just the greenhouse gas emissions aspect. Anyone else got any suggestions as to what areas we should look at? Right, I think it's useful to know what's available from the people who are involved with resources research, and likewise what the ideal feedstock is for people researching conversion processes. And so the link between the resources and conversion catalysis. So yeah, a really important point, Nathan. So generally there's an increase in the number of labs using or wanting real biomass samples. So that's something that we've been looking at, hasn't it, Patricia, in the SuperGen Bioenergy Hub, linking between the resources and conversion topic groups.
Yeah, so there was a workshop carried out on that topic a few months back now, and I think there's a paper, actually, a review paper being prepared based on it. That was more, I guess, around the composition of the feedstock and how that interfaces with the conversion technology being used. But I think Nathan's point is also around the practicality that some people produce biomass and some people actually want it in their labs to use it. And we've had some interesting fun and games in Ebury this week with our biomass samples, yeah, attracting rats, and straw pellets attract rats, just beware of that. And all sorts of interesting fun and games with things going on that weekend. So yeah, that's been great. But yeah, I think if people on the catalysis side certainly are in need of resources, Ian and his team at Aberystwyth regularly harvest miscanthus and other perennial crops. And I think most of the biolabs have access to forest residue and similar stuff. I think we're about to get rid of lots of ours in Aston because we're having a cleanup. So if you happen to be near Birmingham and you need some transporting over to you, we might be able to do that probably this weekend.
It certainly would be worth, I think, if we do have some that have been, that say a crop like miscanthus or grass or whatever it might be, that has been cultivated in different environments, and then do the link between the environmental aspects and what the catalysis can do in terms of conversion, because that, I think, would give us rather than just an analysis of the grass itself or whatever it might be, a link through to what the final outcome would be, because it's not necessarily just the overall composition of the material that matters. It's actually how they're bound together and how easily they are to be taken apart. So if there are things that we can do that have been systematically chained or grown in a particular set of environments, then doing things like hydrogen production via aqueous phase reforming, etc., may be an easy way to try and do that linkage, I think.
Yeah, I think one of the issues there, Chris, is we tend to transport stuff from one lab to another in a nice sort of freeze-dried and powdered type environment because it's easy and it's safe. But the reality is that that's not how people are going to use it, you know. So it's absolutely right.
And actually, when you do those sorts of transformations, in particular with things like lignin, you actually make it harder to do the transformation because you get repolymerization occurring. So.
Yeah, so there's a potential bit of work there, maybe something for flexible funding or something like that.
I would urge people, if there's anything that has come up today that they think they would like to pursue, that there is the flexible funding option for little small projects, and there is the
rapid response.
What do we call it? Sorry, rapid response for small one, and flex funding, which will be in September for the bigger projects.
Yeah.
And, you know, there's, at the peak of this, I think I saw 97, 98 people online. So there's lots of names there of people you can contact. It's not very difficult. And I think that, you know, most people are quite keen to reach out. So it would be lovely to see some joint applications between people in the Catalysis Hub and people in the Bioenergy Hub on topics of common interest.
Yeah, so please reach out to anyone that you've seen talking today or anyone on the audience list. I guess we're sharing the presentations at some point. Nathan or Josie or Helen?
As long as people are happy to share them, then there's no problem about sharing them. Yeah.
And then we've got, everyone's put their contact details on their presentation, so you can just drop them a line. To follow up something that Patricia mentioned at the beginning of the day was about the fellowships, the SuperGen fellowships. So those that will close in December, but we'll be having a webinar in October to tell people exactly what the fellowships are about and the kind of things that we'll be expecting to see in your fellowship application. So stay tuned for some more information about that. If you want to hear about that, then please sign up to the SuperGen newsletter, which should be on the website, and someone could probably type it into the chat about which email they can sign up to. For any early career researchers, if you'd like to join the Share Network, there's another email address to join up to that. Chris, have you got any similar networks within the Catalysis Hub that you'd like to promote?
Not networks. We have each of the science themes, and I've presented those today. And obviously, there's the proposal round that's going on at the moment. So, you know, if people want to put in proposals for that proposal round, then you've got till next Friday to do that. It's only a 2-pager, etc., etc. So that there are networks. We tend to do it in either the linkages with other hubs or via the whole hub. But if anyone wants to if anyone wants to be in touch with people that do X, Y, or Z in terms of catalysis, you know, I've got grass, I'd like to try and produce hydrogen. You know, if you go via me or Josie, then we can put people in touch with each other. That won't be a problem.
And I think likewise, if any people on the Catalysis side need particular bioenergy samples, if they could get in touch with Emma Wild, who's the SuperGen project manager, we can go around our mailing list to see if anyone can provide that.
Yeah.
Yeah. Is there any more questions or comments that you'd like to make?
There's been a couple of things put in the chat from Corinne and Josie about both the proposal round as well as the mailing list for the hub. So, and if anybody would like to go to a conference, it's virtual, and it's virtual in Manchester, if you see what I mean. It's on the International Congress on Environmental Catalysis, which will include hydrogen production and all sorts of other things to do with biomass and plastics. That's being held at the beginning of September. So it's £60 for registration, and if anybody would like to go, then we can then give me a shout. So.
Okay.
Just one other thing I'm going to mention, Katie, sorry. And again, it's from an early career perspective, that one of the things that we try to do in the Bioenergy Hub is also, it was just hearing Chris mention the £60 figure, is we also try to provide support for people to travel to conferences and present things and stuff. So if there are needs around that as well, if you could reach out to us, then we can see what we have in place. I think we used to have a fund that people could apply for on a sort of routine basis. I think we're reviewing that at the moment, and we will issue clearer guidance in September just because of the COVID situation, but there is potentially support available for that.
Thank you, Patricia. Any last words from you, Chris?
No, just to say thank you very much to you, Katie, for chairing a session, and also to Patricia. But mainly, I think we should thank the speakers, the people who've turned up, but predominantly the Josie and Corinne and Nathan and Helen, because without them this would not have happened. So thank you very much indeed to all 4 of you. You've done a great job. It was really stimulating what we were hearing, and it was a good overview, I think, of both the SuperGen and the Catalysis Hub activities. So hopefully this will stimulate some further feedback and research that can be funded through one or the other of the routes that we've mentioned.
Yeah, and I'd just like to reiterate that. Thank you all for organising. The guys, you've done a great job, and also to all of our presenters, brilliant presentations today. And then to all of our attendees, thank you so much for taking the time to join us today. And it's nice to hear from you all over the world. So that was quite exciting. And so thank you from me. Hope to work with some of you all again in the future.
Josie, what are we doing with the recording? Are you going to send around a link to that or something? You're muted. You're muted still.
Sorry, I had my video on, so I hadn't realised I was muted. We'll put it up on the Hub websites, and I'll send it to Emma to go on the SuperGen.
Okay, that's great.
Sites.
Good. Thank you very much indeed. Good.
So thanks everyone. I think that's it for today. I'm hoping we'll hear from you all soon, and that it leads to some exciting work in the future. So enjoy the rest of your day and your bonus 20 minutes.
I know, excellent. Thanks, bye.
Thank you everyone, bye.